This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 654, recorded on August 14th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York State, Daniel Griffin.
Hello, everybody.
And also from New York State, Chuck Knirsch.
Hi, Vincent, Daniel. Good to see you both.
So let me go ahead with our update. I know we have a lot of clinicians, and this has become, initially it was the ProHealth New York, but it's now become the ProHealth Riverside Tri-State Area Clinician Update as we've sort of grown and merged. Let me start with my quotation, and this week it's actually going to be song lyrics, trying to reach out to the younger crowd. And so this is a song, Epiphany, from the album Folklore by Taylor Swift. I don't know, Vincent, if you're a fan or not, but Chuck, you've got some teenagers, so maybe you're familiar with Taylor Swift.
Sure.
So yeah, when I'm not doing this and seeing patients, I'm watching Doctor Who and listening to Taylor Swift, just to give people some background. But so she actually wrote this song, and it came out, and it's about the healthcare workers and the experience with COVID-19. So just something med school did not cover: someone's daughter, someone's mother. Hold your hand through plastic now. Doc, I think she's crashing out, and some things you just can't speak about. Only 20 minutes to sleep, but you dream of some epiphany. Just one single glimpse of relief to make some sense of what you've seen. And I like to put this up front because I just want to try to keep a human face on the pandemic. Sometimes I feel like people lose sight of that, that these, the tens of thousands of people that are affected, the death counts, these each one of those numbers is an individual. And we're not doing well, right? We hit our highest one-day death count since May this week with over 1,500 deaths in a single day. And then there was some data out of the CDC where, looking from March to now, we're seeing over 200,000 excess deaths based on this data. So some even actually say the actual number may be even higher than losing 200,000 Americans. Because I would say it takes time to die, but it also takes time to count all the deaths in the country. So I want to actually applaud in the sort of, in this context here, the Houston Methodist Hospital. I don't know if people saw this, but when we were seeing many COVID cases here in New York, I have to say I was a little bit frustrated, and I know the media was a bit frustrated and would reach out to me because it was really hard for them to enter the hospitals and really report and show people how bad it really was, how overwhelming the situation was. And I understand to some degree the hospitals didn't want to look bad. They didn't want to be the COVID hospital. But I think because of that, a lot of people still don't have a grasp of the human tragedy that was here. But this was an article called "Inside the Fight to Save Houston's Most Vulnerable." It was a New York Times article. And they really, I think they did a great job of really capturing the human impact of this and bringing it to home. And they actually went through several individuals, and they talk about how, it isn't the young people at the gatherings who are maybe not following the rules, but they talk about an aunt, a grandmother, a father, the delivery worker, and all the really hardworking people who are just trying to kind of get through their day and end up getting exposed, end up suffering from the virus. And so I'm actually perhaps a little optimistic that when people read stories like this and it makes the virus real and the danger understandable, that people will start to appreciate where they're being asked to make the sacrifices, and maybe making those sacrifices will become a little bit more acceptable because, these, as I guess in the Taylor Swift song, this is, I think I have an alarm that just went off there. That was my alarm to get ready for this show. But no, I think as people look through this, they'll see, the number, all those numbers, that's someone's daughter. That's someone's mother. This is a human being. Those aren't just numbers. So, thank you to the Houston Hospital for opening their doors and telling these stories. What I'll say also is transmission. There was another preprint out there. I think everybody is obsessed with the aerosol producing procedures and the possibility that this is an aerosolized. And this was a report. It's called Viable SARS-CoV-2 in the air of a hospital room with COVID-19 patient. It was a preprint. And they actually cultured virus and showed CPE. So our TWiV listeners will know cytopathic effect. From what they obtained from the air in the room of this patient. I don't think that changes what we're still saying. We still say, and I think when Anthony Fauci was on, he was very clear that the majority of transmission here is droplets. There's some contact, right? When people touch a surface and maybe they touch their nose or face like Vincent's doing. I'm just watching that as he infects himself with SARS-CoV-2 there.
Yeah, I've been home all day.
And then there are certain circumstances where you can have aerosol. So we know particularly if there's aerosol producing procedures, but we also know, right, that there's a big impact of transmission when people are indoors. And unfortunately, I feel like we're losing our warm weather window to really get the numbers down in a lot of areas. And as people come back indoors, we're going to have more transmission issues.
May I say something about that study?
Yes, please do.
We have no idea if the amount of infectious virus in those aerosols was enough to infect anyone. We know there's a patient in the room who is exhaling virus. There's going to be some infectious virus in the air. What we do know is if you're opposite someone for 15 minutes, that's when you're likely to get infected because you're constantly inhaling droplets. So I'm not worried at all about a few aerosols with virus in them because we have no idea if they're transmitting by that route. The droplets, as you say, are really the key.
I think that's an excellent point because, these preprints, these things end up out there and then suddenly the media jumps on it and suddenly there's certain things I think that we've repeatedly talked about in the, call it the misinfodemic. So it's really misinformation. It's this fear that, every day we find something new and, oh my gosh, it's all wrong what we've been told, but it's not all wrong. I mean, we're millions of cases into this. It's mainly droplet spread. And there's a minority of situations where it might be spread other ways.
I saw headlines in the Times about that article saying, this changes everything. No, it doesn't. Come on, people.
Yeah. Remember, good news is not a headline. Bad news is a headline.
That's right. You're absolutely right.
So testing, actually. So I'm going to try to devote a lot of what we talked today about testing. Because I really think this is key. And I was listening to the most recent TWiV. That's what I also do when I'm not listening to Taylor Swift, watching Doctor Who, or taking care of patients. As I listen to TWiV, which, hey, I'm going to recommend everyone listen to TWiV. But just a little background, because I know some things were thrown out there, Clia, Waived. So I wanted to give people a little bit of context. And then I'm going to say this could be a little bit of optimism, right? I know that my wife refers to me as a bucket of sunshine, not without a bit of sarcasm, but there's a little bit of sunshine in the bucket here today. So when you get a COVID test, when you go in there and you get that swab, that beautiful brain biopsy where a Q-tip goes farther than you knew your nose went back, where did the COVID test? Well, currently about half of these swabs are routed to hospital labs, to university labs, to public labs. And the other half are sent to the 12 major commercial labs throughout our country, such as Quest, LabCorp that we've talked about. But now there's those alternatives to sending off the samples to these licensed labs. And these licensed labs are actually defined, and the Clia Waived are defined by what's called, Clia, what does that stand for? Clinical Laboratory Improvement Amendments, right? So certain tests that we do, we talk about these molecular tests. I like the fact that everything's a molecule. So what makes something a molecular test? But the amplification type technology, the PCR, the isothermic, the nucleic acid amplification tests, these are considered moderate to high complexity tests. And so these have to go to actually certified labs that have been shown to have proficiency in doing that. So these labs actually have to have a Clia certificate. They have to be inspected. They must meet the Clia quality standards. But there's an alternative to this. And I think that we've been sort of on an arc pushing for this. There's what are referred to as Clia waived tests. And these are determined to be simple tests where there's really a low risk of an incorrect result, where there's not a complexity here that it needs to be at one of these sophisticated lab settings. And so these Clia wave tests include the point of care tests that we've talked about quite a bit. Those can be performed in a Clia waived lab or at an office, an urgent care site, or even at a mobile site with a mobile testing license. And these are all tests cleared by the FDA. Also within here are the home use tests, right? So the FDA is sort of telling us which tests can be used where and what hoops need to be jumped through so that we have some reliability with regards to those. Now, if your testing location changes, for instance, you say, oh, we're going to go out to the school or this business, and a lot of times you've actually got to go out there. You've got to get a special mobile testing license. And so I'm going to talk a little bit about how ProHealth is addressing this, because we, like I think everyone else, are looking at the day when everyone has access to. I think Vincent, you want to charge everyone $1. I would like it to be free. So, because I think it's a lot cheaper to have the virus gone than it is to keep dumping $1 trillion to help the stock market go up. But we actually went through like a whole thing this week talking to the tri-state area physicians, so the recommendations for how we do this. And in the 50 urgent care centers in the tri-state area, I think you'll like this, Vincent, we are investing in point of care rapid testing where we'll be able to have results in less than 10 minutes at all our 50 sites. So we're buying all multiple Abbott ID Now machines for our multiple sites. We're in negotiations with BD to get those rapid lateral flow tests. And so what we're really trying to do is actually is when people say, hey, I can't go ahead and open up my school, my business with a testing as part of the program, we're saying, give us a call. We'll make it happen. Mm-hmm. And when those $1 tests come out, that's great. But those $1 tests are not here today, and you're planning on opening today. So let's make this happen. So just to let people know, the rapid getting a result within minutes it's here and it can happen. Anyone who says, "Oh, we just can't make this happen," there's no excuse. Just to make that public.
And you'll report the results, Daniel, the positives particularly, but also just to know what our percent positive trend, because I think that information is useful to public health, Department of Health.
Yeah, all this stuff is tied in, Chuck, with the Department of Health. So I think this week we actually in New York State we got up to the point where we are doing over 80,000 tests a day. I mean, so we are, there were two responses. One was, oh, there's resulting delays, we should test less. Our response was we should fix the resulting delays. Let's get that resulting delay down to, so now we have it down to minutes in several places. And the worst we're seeing is 36 hours, 36 hours is much, much better. That's having a test. As Anthony Fauci was saying, if it's 8 days, if it's 13 days, as it is in certain states, that's like not having a test. Mm-hmm. So let's all do this. Let's make this happen. I think there could be a political will. There could be an economic will. This could be done. We're doing it. We're making it happen. So if you're not making it happen, then figure out what's in the way, because we need testing. I'll get back to testing. I won't be able to leave it alone for the next little bit. Masks. There was it wasn't wonderful. There was an article that came out in Science Advances, low-cost measurement of face mask efficiency for filtering expelled droplets during speech. I don't know if everyone got a chance to see that. Mm-hmm.
Yeah, I did.
It was sort of painful. It started. I'm not happy with the gator shaming that resulted from this article. I mean, there were some things that were interesting in there, but I'm not sure this study carries as much weight. I mean, there were certain things that looked interesting: the N95s, the surgical masks, the cotton masks that a lot of people are wearing looked pretty good. It was an interesting article. Vincent, do you have a take on this at all?
I think they measure droplets by laser. I'm not interested. I'm interested in which droplets have virus and what the masks do. And so, in my opinion, this study is useless and should never have been published.
I'm a little worried about the study, too, because one of the authors was on, I forget what TV show I was watching late last night, and was suggesting that large droplets could be converted into aerosols with a single-ply gaiter. I really, think that's a bad message to be putting out. And I'm glad to see our virologist shaking his head, because I agree that these things shouldn't, get published.
Yeah, I mean, it's worrisome because once this gets published, right, then everyone sort of jumps on it, and you see all these, and I was seriously, you're seeing in social media now gator shaming, where people are being tagged, with pictures. I saw your kids wearing gaiters. According to the latest research, that makes it worse. Somehow wearing a gaiter magnifies the amount of virus that comes out of your mouth. And so hopefully, again, this is our countering the miasma of misinformation. This doesn't change everything. Wear
masks. Wear masks.
Keep wearing. Yeah, wear masks. School opening plans. I was going to skip this, right? I was going to not talk about school reopening plans, but you can't skip it. People say, you're not going to talk about it? So I got, in the group of infectious disease doctors that I work with, one of the questions that came up actually earlier today was, we have a lot of individuals who have children, and they have, they're older in age. They have other medical problems. Maybe they have lung disease, heart disease, and they're worried. Maybe they even have immune issues, and they're worried. If my kids go to school, what will happen to me? And and we're seeing now plans in our area where testing is not part of the plan. They're just going to send the kids, much like Georgia, they're going to send them off to school. They're going to be together even in a school bus, for instance. They're going to be together in a classroom. And then we know what happens. I mean, schools, these are cruise ships on land, and they're going to spread the virus, and they're going to bring it home to these individuals. And the individuals are now asking, what can we do as physicians to guide them with regard to the kids going back to school? So, this, like many listeners, this impacts me very personally. According to the American Academy of Pediatrics, the change in child COVID-19 cases from beginning of July to beginning of August, right, over that period of time, we had a 90% increase in the number of cases in children in that 4-week period from the period before. I mean, just to be straight, kids can catch COVID-19. There's every reason to believe that kids can spread COVID-19. So what I worry about it, and my comment to this group was, opening your schools without a plan, without testing, is basically the equivalent of saying, go out there and drive. But we've taken away your brakes, we've taken away your seatbelts. And by the way, those people with comorbidities, we're going to pack them in the front seat blindfolded, and we're going to let the children take the steering wheel. So, as we just saw the experience in Georgia, I was really, I guess, astounded by the different media takes. Some people said, see, we picked up these positive cases, we shut the schools, we have a plan and it works. I was like, I don't consider that working, but okay. If you're okay with lots of kids getting sick and then bringing it home to their parents, open your schools without testing. But as I started with, there are groups. I mentioned Quest, LabCorp, ProHealth. There's a lot of people with the ability to test. Let's work together. If we're going to make this happen, and I feel a certain pressure. My daughter, Eloise, my middle daughter, who won the sailing race, she told me, Daddy, if they open the schools and you don't let me go, I will hate you. And, no one wants to be hated by their child. So.
Daniel, I don't know if you saw this week in the Times, there was an op-ed by a pediatrician who claimed that children don't transmit. And I just do not understand how she can say that when we know historically children drive pandemics. There's no doubt about it. Every pandemic, school and children drives transmission. So I just don't understand why she is saying this. Do you?
I mean, every respiratory I look at the school calendar and determine my vacations. I don't take off when there's a school break. I basically prepare that right after the school breaks is when I'm going to really be busy and I need to be around, because whether it's influenza or whatever respiratory virus, when kids go to schools, it starts circulating. And then when they go spend time with family, what did we teach them in kindergarten? We taught them to share. And so we teach them to share in school, and they certainly do that with respiratory pathogens.
It's not just children. I mean, when office buildings open up, if they don't have a plan to test and to use masks religiously 100% of the time indoors, So I think it's the congregation that is important. I think, some of the, immune changes with influenza, I don't know if we can port that to SARS-2 yet, but I think, we're thinking a little bit about that. So testing has to be part of the school plan. And the universities they're trying to open have been doing the test runs of having, different sites around campus, but they're also doing, food to go, not sitting around the dining halls. And so school will look drastically different for those that are trying to open, and hopefully this will be successful. But it is an experiment right now.
Yeah. Yeah. I mean, it's an experiment with the high cost, right? Experimenting with the lives of our children and families. But I like that you bring up also work, because we also get questions about that. We're in the tri-state area. At the center of this tri-state area is Manhattan, with these tall 50-story buildings. And how do people get from the lobby up to their office, right? Elevators. And in residential, they'll do things where it's like one person or one family in the elevator at a time. Just imagine trying to get to work in a city where there's a limited number of elevators. By the time like everyone goes in one at a time, it's 5 o'clock and the lobby's still full. So we really need, testing to be part of this, so that we can open the economy and do these things safely. The other is I'm going to give people homework, right? So this is a teaser for next time. This is back to evidence-based medicine, and next time when we meet as my prep, I'm going to go through for the clinicians. What have we learned so far at the different stages, and also how have we learned it, right? Because it's been a little bit back and forth here. So back on TWiV 606, that was released in the end of April 26th, I discussed the different approaches that physicians might use when they're selecting therapy for their patients, right? We talked about eminence, vehemence, eloquence, diffidence, nervousness, experience-based, and then finally we got to evidence-based. And there was a New York Times article called The COVID Drug Wars that pitted doctor versus doctor, published August 8th, 2020. It's actually a very long, detailed article. And I thought it was really interesting. It described the conflicts around these issues at the Northwell Hospital. So a lot of these people I know personally. And it's not an easy read, but I think it's an important topic as we sort of continue to move forward. Because here we are, maybe 6 months into the pandemic here in the New York area. And we're still limited in what we have as far as randomized controlled trials. But I got to start asking people to listen to that TWiV, at least the first part. You can keep going if you really get hooked. Look through this, because in the next episode, I'm going to talk a little bit about what we know and how we learn this in this paradigm. The long haulers, I just want to say a couple things about this. One is I was just on the phone earlier with a young woman, and the loss of taste and sense of smell has an interesting clinical course, I'm going to say. Like this is actually quite upsetting to people, where they lose their sense of taste and smell. But then what happens is something that comes back, sort of this foul smell, this metallic smell or taste. Interesting enough, that looks like it's a precursor to people getting better. So we're starting to have enough experience now that I can sort of say, oh, okay, you're now getting that metallic, that foul. This seems to be things coming back. And it looks like then they're going to get the taste and smell back. So that's a bit encouraging. It's a little bit of sunshine, right? When you get that nasty smell and taste, that's encouraging. The other, I wanted to talk about long haulers and children. There's been a bunch of articles about this recently. And again, this is going to be reassuring. I was talking to Jay Berger, who is the head of our pediatrics, chief of our pediatrics group. And we have thousands and thousands of children that are under the care of the ProHealth pediatric department. We are not really seeing much in the way of the long haul in the children. This is a rarity, fortunately. So children can get infected. Children can transmit. They are less likely to get severely ill, and they are less likely to enter into the long haul, the tail phase of COVID. So I'm going to finish on that note, and I'm going to thank everybody for continuing to help us support Floating Doctors, right? We switched our charity, and Floating Doctors is the group that I work with down in Panama that helps the indigenous people in the Northeast archipelago. And as I mentioned last time, they've been for years going out to all these little villages by boat and helping these people, setting up mobile clinics. And they're continuing to do that. But now, due to COVID-19, there's incredible challenges with food insecurity, and the people are literally starving. So help us raise money. Go to parasiteswithoutborders.com and donate, and we will double your donations. We're hoping to get that up to $40,000 that we can donate to Floating Doctors to help feed these really hard-hit individuals and continue to provide medical care.
Very good. So, Daniel, I wanted to ask you, you had sent me a document about testing. Do you want to put that on the show notes for today, which has your table, your COVID test table, RNA levels, infectivity, and so forth?
Sure. Let me do that. Yeah, I'm going to send it to you again because I left like a little thing out in the table. So yeah, and I guess for our listeners, we've had a lot of people ask about, hey, how do I get that table where Dr. Griffin talks about different viral loads and the sensitivity of different testing modalities. And so it's in there where we talk about different RNA copy number levels and really the point at which people become infectious, as well as which tests can give you a positive result at different levels.
Great. Yeah, we get a lot of requests for that. All right. Also, both of you, Daniel and Chuck, what do you think of this MBio paper? Is it MBio? I thought it was. Yeah, MBio came out this week. Effect of convalescent plasma on mortality among hospitalized patients is the first 3 months in this very large study, which is not a controlled trial, right?
We were part of this.
Mm-hmm.
So I certainly had patients enroll. And just to give a little background, and I'll let Chuck jump in as well so we can go back and forth. But what this was really a compassionate use type of protocol.
Mm-hmm.
So you could just say, hey, we now have access to so many, batches of this plasma, and you would go through and just give it to a whole bunch of people. And so there were a bunch of issues with this as far as the publication. So it was not in any way a randomized controlled trial.
Mm-hmm.
But then what they did is they went back through this. I actually found this publication, I'm going to use the word offensive in their discussion. They're a little too happy patting themselves in the back. I think some shoulders got dislocated. But what they say in their discussion is when they set this up, there were a bunch of randomized controlled trials out there where individuals could be enrolled by their physicians into either a placebo arm or a convalescent serum arm. Or instead, they could say, you know what, I just want to give my patient convalescent serum because we all believe it's true. And they offered the wonderful opportunity to just circumvent the randomized controlled trials and just give everyone convalescent plasma who may or may not need it. And then they show a couple things in the study. They show that, early on, people weren't doing so well. And then as time went by, the outcomes were somewhat better. And then they proceed to say a couple of things. They said, you know what? The earlier in the course of disease that people got the plasma, the better they did. Oh, and by the way, as time went on, people were getting it earlier in the course of disease, whether they got convalescent plasma or not, then happened early on. So if you look at it, back in March and early April, people were not doing well. And you didn't have a lot of access to convalescent serum. So people would be sitting in the ICU for a month. They were not doing well. You didn't expect them to do well. The plasma would show up and they would get it. By the end of the study, as we saw, the plasma had higher levels of IgG. People usually were not quite as sick, but we had better access. So less sick individuals were getting it. And they found out, surprisingly, that the people that, the way I interpret the data, the people that got the plasma later on, earlier with higher IgG levels, did better than patients that got the plasma early on, back in April when people weren't doing well. So I really thought it was, in a sense, a terrible study because it's going to give support. But what they really did was, in a sense, they cut the heels out of people who are trying to do proper randomized controlled trials. And here we are in August, and we still don't really know whether convalescent serum works. And if anything, there is one randomized controlled trial out there that showed no benefit. Mm-hmm. And there could be more if they hadn't had this quote unquote compassionate use program.
Right. So
I'm now going to step off the soapbox. I'm going to let you step on it, Charles.
Well, so this is a resource intense procedure, right? And not without risk. And so we actually need to know whether it works. And so I look at it in terms of the weight of evidence. And I would place this in the category of big data observational studies. interesting, but what is hypothesis generating for the randomized controlled trial that must be done. So it would not alter, my clinical practice, as it's clearly true, what you're saying as well, but it would have me design a randomized controlled trial. And if this did undermine randomized controlled trials, that's a shame. That just shouldn't happen.
Yeah, I mean, they really, Vince, like if you look through the discussion, they sort of pat themselves on the back saying, a lot of doctors would rather just give what they think works than enroll their patients in a trial. And we gave them that option. And so I was sort of like, oh my.
From my view, I mean, these patients got all sorts of other treatments in addition to the serum. I mean, it's all over the place. And if you look at the graphs where they're comparing recovery with, doses and so forth, all the error bars overlap. So you could argue that they're really all the same. And so would this change what you do with patients, Daniel? Would you give them serum based on this?
No. I mean, if anything, this was a huge data set, with, many tens of thousands of patients, and they still couldn't show anything significant, even with all the flaws, which would sort of bias it towards showing something significant. So if anything, I see it as more sort of science that does not get me excited about convalescent plasma. I mean, if you need more than 10,000 patients in a study and you can't show significance, I'm not really excited. And they do they say, 30% whatever reduction, but 30%, if you take like 12% and move it down to 10, I mean, it's,
it just seems
the
to
absolute
me that
numbers are very small.
That when you're in the hospital, you're beyond the virus stage, right? You have other issues that aren't going to be mostly taken care of with antivirals like this. We see that with remdesivir, right? It has a modest effect because what you need is modulation of your immune system, et cetera. Okay.
Yeah. And I think we're going to have engineered antibodies, that have been properly studied because they have to be studied, that would replace this anyway.
I'd be surprised if they have much.
If it works.
I'll be surprised if they have an effect because, again, hospitalized patients, IV, I think is going to have a modest effect, but we'll see.
Yeah, no, I agree. I think I talked about earlier that United Health Group was trying to partner with organizations that make the monoclonal cocktails to give them to high-risk patients in urgent cares in that first week when they should work, rather than waiting until the virus is mostly gone and you're in this cytokine storm stage.
Okay.
Yeah.
Dan, you want to do a couple of emails before we break?
Oh, certainly.
All right. Paul, our COO of our hospital system contracted COVID-19. He had cytokine storm, multi-organ failure, respiratory failure, ventilatory support. He did receive convalescent serum. This was early March. He recovered, was discharged. Now he has lymphoma. You've spoken how there's some thought that the virus might continue to exist or hibernate in lymphocytes. EBV is known to cause Burkitt's. Many other viruses can lead to malignancies. Question is, are you seeing an increase in lymphomas in COVID-19 patients who recover?
Yeah, so Paul, thank you for this email. You hit several interesting issues here. One is this question about people get infected with COVID. I should say, people get COVID-19, they get infected with SARS-CoV-2. And then there's a whole question about when does the virus completely clear from the system? And is there some persistence or whatever we want to call it? I don't want to use the word latency because there's no genes that would actually establish latency. And I think Vincent probably, hopefully you'll echo that or not.
I agree with you. Nothing we know.
Yeah, but latency is not the right word, but it may be some delayed clearance. We had an interesting case recently that really played into this and makes me a little worried. It was an individual who had COVID-19 early on. They were a lung transplant patient. And when they started to have issues with rejection, the medicines were increased. So their immunosuppressive medicines were increased. And when they did that, the person started to actually seeming to get sick, looked like they had developed acute COVID, but we knew they had COVID back in March, so it didn't make any sense. Did a COVID PCR and it was positive. So they had been sick. They had had many negative tests, right? Because we did that before they were put on immunosuppression. And now they actually, so I'm a little bit concerned about this whole concept of delayed clearance, as we've seen in certain things like Zika. Not that there's any latency, and I don't know if it's hibernating in lymphocytes. I certainly don't know how exactly it's persisting, but that is a little concerning that when people have a drop in their immune function, that maybe we're seeing controlled virus that's not completely cleared, maybe potentially start to replicate again. But as far as lymphomas, this is actually an area that I care, I guess, deeply about. I'm hoping I can eventually get funding to get back to finish what I was working on before COVID-19. But we saw a significant increase in lymphomas in patients with HIV, even patients that were well controlled, that had undetectable viral loads, that had good levels of T cells. And we know with Burkitt's lymphoma, there's some sort of chronic inflammation that we poorly understood. I always feel like it's hand waving when I say chronic inflammation, but some sort of immune activation that looks like it links certain infections to malignancies, particularly lymphomas. I don't think we have enough time to really see this at a significant level during this pandemic, but time will tell. I haven't seen it myself. I don't know if there's enough time to go by, but this is definitely something I'm going to have my eye on. Is do we see like post-COVID complications that would fall into this category?
All right, Bob wants to know if you have seen COVID-19 patients with POTS. And if so, what can be done? What's the prognosis?
So we've certainly seen people develop what might be POTS, what might be this postural orthostatic tachycardia syndrome. And there was a Lancet Neurology article that came out, neurological complications. Actually, there were a few patients in there that looked like they might fit this criteria, where they develop COVID, and then afterwards they're having issues when they try to stand. They have tachycardia. They have problems maintaining their blood pressure. So we don't really know. But I would say early on we're seeing what looked like an autonomic hit. So just to give people a sense, we have our sympathetic and parasympathetic autonomic systems. So when we stand up, this allows us to maintain our blood pressure. And a lot of people after COVID in this tail phase seem like they're having issues with hypotension, with, we'll say, autonomic dysfunction. I don't know if they're going to actually fall into a classic POTS syndrome or not. I'm still hoping these people eventually get better. But yeah, we're starting to see some reports of this.
Okay. Liz wants to know, she has two questions. First, cytokine storm and tail, the long haulers you're seeing, is this unusual for a viral illness?
So it isn't actually, and I think we've talked about this on TWiV, is a small percent of patients with other viral illnesses, we'll use influenza as an example, have a tail. We know chikungunya is another classic where people get this viral syndrome, and 3 years out, 10% of people still have severe joint pain and fatigue. I actually have a friend, a physician in Seattle, who, 3 years still having like arthritis and fatigue and issues. And he was, so yeah, unfortunately, certain viruses have this tail. It seems to be more common. It's either more common in COVID, or there's just so many more cases that it's really sort of standing out for us.
Okay. And she has a reason for asking that, because she has a freshman who had similar symptoms but didn't have COVID.
Yeah.
All right. Secondly, do you believe that the centers studying COVID long haulers will be able to help them suffering the effects of other viruses in particular?
Yeah, so I hope, Liz, that we learn a lot. And that's one of my optimism, the silver lining of the pandemic. I feel like we will learn a tremendous amount. I think we already have learned a tremendous amount. So I do have a lot of optimism that centers that are studying the COVID long haulers will be able to help not only the COVID long haulers, but that we're also going to be able to help other people that have post-viral effects of other viruses.
Yeah.
So yeah, I'm quite optimistic here. Yeah.
All right. One more.
I guess I should.
Yes.
I was going to, before you go there, this reminded me of, I think, I thought it was a great case. Everyone can tell me if it's a great case, but it was early on in the pandemic. This was in March. And I admitted a woman who had typhoid, enteric fever, right? And one of the classic things about that is when you get a fever with typhoid, your heart rate stays low. I don't know if people have noticed, but normally when you get a fever, you get a rapid heart rate. And when this woman was getting better, everything was great. It was a very interesting story. She had a cousin visiting from Pakistan who actually had multi-drug resistant typhoid and was spreading it to all the family members that we eventually figured out after talking to her. But when she came in, she initially had that high fever. She had the low heart rate. She was getting better, but she was getting better in a hospital that was starting to fill with COVID patients. She then developed a second fever, and then this time she had the high heart rate. So normally, COVID fever, you have a high heart rate associated with the fever. You usually don't have, at least early on, the slow heart rate when you're having those fevers. So,
all right, last one from Amal, who lives in Paris. He's working and he's out and about. He takes precautions. He's worried because he's on anti-inflammatory drugs for shoulder tendinopathy, and he's worried if he won't get symptoms as a consequence and he won't know he's infected. Should he be worried?
Yeah, it's interesting. I was reading through this, and so I work, so I take public transportation, and I also go to the gym every day. So I'm trying to figure out if going to the balance between going to the gym every day and exercising relative to the risk of going to the gym and exposing oneself to other people who are at the gym, breathing and doing all those things that people do. What I will say is the anti-inflammatory drugs, early on, there was sort of this big scare. There was a letter to Lancet, and it was basically, we're a little concerned about anti-inflammatory medications and COVID. That has not panned out. If anything, sort of a shame, because the anti-inflammatory medicines like Aleve, like ibuprofen, like naproxen, really can have a tremendous impact on the symptoms. I mean, when we finally realized, like, hey, it was okay to use these, and you would say, well, why don't you take, an Aleve before bed, a naproxen before bed? Why don't you take a few ibuprofen? People are like, oh my gosh, I felt so much better. And we felt kind of terrible that we had deprived them of this relief, because as we know, sometimes people have COVID not for just 2 weeks, but 3 weeks, but 8 weeks. And the fevers tend to come in the evening and at night. And these nonsteroidal anti-inflammatory drugs can actually really make a big difference. So I'm not particularly concerned, Amal. I think it's okay as far as those go. But yeah, don't touch your face, wash your hands, keep your physical distance, do all the other things. But I don't think that these nonsteroidal anti-inflammatory drugs are going to shut down your immune system.
All right. That's our weekly COVID-19 clinical report from Dr. Daniel Griffin. Thank you, Daniel.
Oh, my pleasure.
And with commentary by Dr. Chuck Knersch. Thanks, Chuck.
Good being with you both.
Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 87 and pretty humid here.
From Western Massachusetts, Alan Dove.
Good to be here. It's 86 Fahrenheit, 30 C, and the dew point's 58. So it's actually kind of nice. Blue skies.
From Austin, Texas, Rich Condit.
Hi there. It's actually now it's 99, headed for 105.
Wow.
Blue skies.
Wow.
Oof. Oof.
It's like 115 there, right?
It is, yes, 115.
And
we're not at the peak temperature yet.
And from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent. Brianne, what she said, that's what it is here.
Yeah, I forgot. 87, I think.
87,
yes.
Humid. In the Catskills where I do my fishing, and in the Poconos where I do my fishing, for the last 3 weeks, no river has failed to reach 70 degrees during some time of the day. And that's the temperature that drives all the fish into the spring hole. So I am fishless.
Fishless.
I'm fishless in Seattle.
If you go to the Poconos, can you then come back to New Jersey without a 2-week quarantine?
Nobody's saying anything to me when I go to my club.
Is New Jersey doing that?
New Jersey is.
No,
I don't know about
know.
Pennsylvania's rules.
But I have a Jersey license. I have a Jersey driver's license.
The thing is, if I go to Pennsylvania right now and come back, then I am supposed to quarantine for 14 days in Massachusetts and not not leave the house at all.
I
and
haven't heard
not
this.
have-
I haven't
yeah.
heard this.
Oh, they've got signs on the highway advertising this fact. They've got the defined zone, which is basically New England plus New Jersey and New York. I can travel in that zone and not have
But not
quarantine. But if I
Pennsylvania, I
go
see.
outside that to any of the other states that are not on the
Gotcha.
approved list, and the approved
Gotcha.
list is based on
You're going to get email, Dixon.
Well,
Someone's
then
well, but
calling
the
back.
streams are too warm for the fish anyway, so you're not going to the Pennsylvania.
let them say what they want.
I was able to go, Dixon, to my parents' house in upstate New York. New York is on the
New
list,
York's okay. Yeah.
so the Catskills are fine.
Yeah.
Catskills.
No,
are-
the Catskills are cool. That's right, and they are cool, by the way.
and
They will
the
be cooling down.
Adirondacks would also be good
Yes,
and might
they would.
actually have cooler streams.
That's right.
So
They will have cooler streams. You're right.
So
absolutely
today we had a first. We had a guest not show up for the first time ever.
Yep.
So, as a consequence, we don't have Brevia for you. We'd normally have some Brevia, but it's okay. We will go right into some email.
You do knock jokes.
No. But before
Let's
that,
do email.
Let's
before
do it.
that,
Do we have an announcement about a meeting? or
let's
is
have
that-
a meeting. The European Society for Clinical Virology, ESCV, their meeting this year on September 9th, 2020 will be, of course, virtual. And last year I went to that meeting in, where was it? Copenhagen. Very nice. And this year it will be online, of course, and it will be all about COVID-19, bringing together a worldwide community to discuss the pandemic. We'll put a link to the website. Attendance is free of charge. Everybody can go for free. And all you need to do is register. So we will put a link to the registration there, and you can see the program and decide whether you want to go. Should be cool.
There are a whole bunch of horrible things about this pandemic, but I really like some of the things that it's done to scientific meetings.
Mm-hmm.
I mean, the accessibility of I don't want them all to go all online forever. I do want in-person meetings to come back again after we have a vaccine. But the accessibility of something like this, I mean, that's awesome.
No, that's good. It is good. I agree.
The faculty here this week got an email saying you should come back to work at some point.
Why? There's nobody there.
No, there are people here. I mean, this floor, there have always been people here. It's just that they're not as many as before. But I think the wording was, while we would all like to direct our labs from Tuscany or Napa Valley, it's probably not a good idea.
Oh, is that what you think we're doing?
I have been coming in twice a week for several weeks now, and that's fine for me. He said a few days a week at least you should start, because things work better when you're there, and it's true. I have no problem with coming in, as long as we're safe about it. I mean,
Exactly.
We've had some construction going on here on the floor for a future tenant, and the construction guys are not always so careful. I don't really appreciate that.
That's right. Yeah.
Okay, our first email is from Brian, and he titled it "Frustration." Dear TWiV team, as a school board president of a small California district, I contacted Kidel to learn about their Sophia antigen tests. It requires nasal swabs. They're not available to schools until 2021, but I doubt a school can run nasal swab tests. I would agree with that. I think saliva is the way to go.
Yeah, I did.
Kidel's representative wrote me that, quote, current FDA language says that a negative test should be reflexed to PCR for confirmation. There's no language in our package insert that refers to transmission, end quote. Yes, of course not. That's too forward thinking, right? I'm not surprised at all. And the use of the word reflexed, come on, that's not common language. Give me a
It's not
break.
English.
Plus the fact if all the negatives have to be confirmed by PCR, it's useless.
No,
Yeah.
the point is you have a rapid test you do every day. You do it the next day. And if it's negative,
Right.
you're probably below the transmission rate. But obviously this hasn't percolated, right?
No.
All right. So back to Brian. This is the problem. FDA says they have to be referenced to PCR, and transmission is not considered relevant. Yes, I totally agree with you. We're not there yet. Doctors tell me the cheap, fast tests are inaccurate. When I explain they're insensitive, not inaccurate, they say, but what about false negatives? So
Right.
not only has the FDA not got it, but neither have most doctors in this country. People like Daniel Griffin get it.
Well, the doctors are going to be the last to get it. And that's fine.
Absolutely.
That's fine. They don't need to figure it out until it's in general use.
But really, frankly, the doctors are not driving what kind of tests you're going to be doing, right? That's going to come down. It's going to percolate down with them. But so this is totally not surprising that doctors don't get it, except for some.
Those doctors who listen to TWiV and Daniel Griffin, I'm sure.
Yes, so Daniel and Daniel's network who are listening, you get this. It's got to percolate beyond, and that's a challenge. It really is. I understand. Back to Brian. Accuracy means a result represents an ontological value. A cheap test report, you have no more than 50,000 virus particles. PCR accuracy states you have no more than 999 virus particles. But even academics fall into the trap of pretending precision is accuracy. How many papers report statistical results to the third or fourth decimal point when the next experiment may have results that differ by a whole number? Okay, yes, but it doesn't help in this situation. FDA is unwilling. And in fact, I would gather that many scientists don't get this. Accuracy isn't important either, right? It's tough to do. The FDA is unwilling to make the effort to explain testing for transmission, even if they make noise about inexpensive tests. They're afraid of the Atlantic, the New York Times, the Post, CNN, CBS, etc., all of whom will run headlines about inaccurate tests. They're afraid of grandstanding politicians. They're afraid of an unpredictable, ignorant president. They're not afraid of scientists or educated citizens. Yes, I think you're right. And finally, game theory. And we have two people in gaming chairs, so you tell us if this is right. Game theory says the best option is rarely chosen. If people are clever, they settle for second best, so I'll accept pooled testing. How frustrating, Brian. Is that true? Game theory says the best option is rarely chosen? Oh, you guys are not gamers, really, right?
No, well, and game theory is separate from gaming.
True. That's true.
So, game theory is a whole discipline, and I think I get this idea of the best option rarely being chosen because usually the best option involves some big expense or trade-off that makes it, makes more sense to choose the second best option because that's going to be quicker.
The sad thing is, in this case, the best option is cheaper. But it doesn't resonate with people for reasons that you are hearing, and we have to keep working. And so that's why we've seen a little lull in the excitement about letter writing. Got to move back to that. I know a lot of people think it doesn't work, but anyway, sorry, Brian. I get it. We get it.
Yeah.
It's very frustrating.
Yep.
Alan.
Anne writes, Dear TWiV, I was turned on to your podcast by TWiV 640 featuring Michael Mina and have since been an avid listener and learning a lot. I've even written my first ever letters to public officials as a result of that podcast, and now I'm writing my first ever letter to a podcast. So somebody's writing letters. As a pathologist who has dealt with bringing up the SARS-CoV-2 PCR assay at local hospitals, I would like to point out something I haven't heard you mention. Specifically, there's a difference between analytical and clinical sensitivity. Analytical sensitivity can be described as the technical ability of a test to detect a small amount of analyte, but clinical sensitivity has to account for how the test can actually identify disease with real patient samples. Unfortunately, there really aren't a lot of data, at least that I can find, about the clinical sensitivity of the PCR test, but depending on the source of specimen and when in the course of infection it's collected, unpublished data suggests a clinical sensitivity range of greater than 90% to less than 70%. I think this distinction is important because patients that have tested negative by PCR but have a clinical presentation consistent with COVID-19 should probably be tested again or get the serology test despite the high analytical sensitivity of the test. In regards to how we bring up clinical tests, in TWiV 651, Dixon mentioned that samples are sent by the College of American Pathologists to local clinical laboratories. But I think he was actually referring to proficiency testing. The CAP test does send out test samples to subscribing clinical labs, but this is for tests that are already validated as regularly occurring quality check.
Yeah, that's right.
Getting samples to validate our SARS-CoV-2 PCR tests was not easy, especially when infection prevalence was low in our area. This was quite a contradiction, as we needed to keep as we needed to test to keep viral spread low, but because it was low, we didn't have samples for test validation.
Catch-22.
Yes, the local health department had to send us patient samples, and we used contrived samples as well as with material obtained through the CDC. Getting samples for test validation has continued to be a challenge with testing asymptomatic people, as the FDA says we cannot use a test to screen asymptomatic individuals unless it was specifically validated with samples from such people. As you may imagine, such people are hard to find when the prevalence is low. Although we recently have found a few such individuals, perhaps reflecting increased local spread. The reality is that many, if not most, labs use the test for screening asymptomatic individuals, as such testing is required by local and federal public health mandates to screen people before medical procedures or for travel. For now, if you look at the FDA emergency use authorizations for PCR tests, only one lab has approval to use their test for asymptomatic screening. But I hope the move by the FDA to distinguish between diagnostic and screening tests will help those of us in the clinical laboratories caught between contradictory regulations and requirements. Thanks for allowing me to share my frustrations and for your ever-enlightening discussions.
Wow.
Thank you, Anne?
It's complicated.
Yes.
Yes, it is.
It seems more complicated than necessary.
Yeah, that part about that part about asymptomatic screening is very interesting. Yeah,
there's a couple of catch-22s in there that there ought to be a workaround.
Yes.
Or not a workaround, but the authorities ought to sort of get on board with this.
Especially now in a pandemic situation, right?
Exactly.
We don't have a lot of time.
Well, specifically now, I mean, what we're seeing here is the impact of rules that are a great idea when there's not a pandemic.
Yeah.
Right.
Yeah.
You don't want unvalidated tests running in a lab, and so you insist on validation. But then we need to rearrange things a little bit given the current situation.
Well, it would be nice if we could fix a lot of this during this pandemic. But even if not, I hope somebody's taken good notes.
Yeah.
So that when we have a scientifically minded administration, we can put together a pandemic preparedness effort that takes all of these lessons into account. So that the next time around, and there will be a next time, we've got procedures in place to deal with this better.
Well, as I've said before, I think when the next pandemic comes around, we will be exquisitely well prepared for this pandemic.
Just like we are currently very prepared for an HIV pandemic.
Yes. If HIV breaks out again, we got it. But we're on that.
I guess you're being sarcastic.
We'll be ready for the next. So SARS-CoV-3 is not going to catch us off guard.
So that's SARS has sarcastic as part of its name, right?
Yes, right.
Got it. Dixon, can you? By the way, Anne has references here for people who want to read about this.
Yes, that's right.
Dixon,
Lizzie writes, "Dear TWiV team, it's 24 degrees Celsius, 76 Fahrenheit in Draper, Utah, and I can see an occasional falling star as the Perseid meteors showers from my roof, even in the city. On even one of them, and in fact in Salt Lake City, there are no bars open because there are very few bars, actually. On every one of them, I wish that phase 3 trials of this new Russian vaccine weren't going to be tested population-wide, starting with the healthcare workers who probably know it's a question in a bottle instead of actual protection. Another concern I have is, how do I explain to my friends and relatives that we are expediting because we have to with this vaccine, and ours will be good, and you should get the one that's approved by the FDA, but Russia is bad, and you should not call your Congress critter, I like that term, and tell them to buy Russian vaccine because they went too fast and didn't show their work." How do I explain this to people? I am still trying to convince not to eat out at restaurants and go to movies at a megaplex. And that's right. There's a whole bunch of good things here.
Good time to talk a little bit about this Russian vaccine quotes.
Yes.
Yeah, tell us about it.
They were Russian, all right. So the thing to know,
that's a great word for that.
Yeah, as I said on Twitter, Russia did not approve an effective vaccine. They approved an effective vaccine. It's been tested on 76 people.
That many?
Yeah, we have virtually no data reports or any kind of transparency on how the trials were done. And this is an adenovirus vectored vaccine, which is not inherently bad, but there are some issues that you'd want to see tested and looked at. Like if people have previously been exposed to adenoviruses, which we all have, they may have pre-existing immunity. The vaccine may be less effective and so forth. So in theory, it's a vaccine that could work. The testing on it so far has been very brief. It's up to what we might consider a phase 1-2 type of trial. And the authorization that it got was an emergency type of approval. It's kind of analogous to the FDA's emergency use approval by the Russian equivalent of the FDA, which, of course, I'm sure was under a huge amount of political pressure. And even with all that, they gave it this emergency approval with a stipulation that it not be used on the general population until at the earliest January 2021. So it is not, in fact, being rolled out to the general population.
Yeah.
The manufacturer of it claims they can make about 1 million doses a year. With their current capacity, yeah. So we're not talking about something that's being ramped up to the scale it would need to be yet. And we don't know if this thing works. And so...
So, Alan, why did Putin announce this?
Because, I mean, why does Trump announce stuff, right? You know, it's hype. It's pure hype. It's propaganda.
So
you
in
think,
a way,
hey, we got our vaccine already.
Yeah.
No, you didn't.
In a way, this sounds almost the same as the discussion we had with the Chinese vaccine to be distributed or made available for the military. It's almost in the realm of a phase 3 trial.
Except that, in this case, it's
not a controlled trial.
I mean, in the case of the Chinese vaccine, my understanding is that they actually have one and that they are, in fact, capable of manufacturing it at scale and that they are, in fact, proceeding with testing at a larger scale. This thing is still, I mean, it's behind the Moderna vaccine. It's behind the couple of others in the Oxford it's definitely behind the Oxford vaccine. And it has not,
and Pfizer too,
and it has not been approved for general use.
So you think?
Go ahead.
Do you think Trump is going to push hard for the Russian vaccine?
Oh, sure. Yeah. I mean, if it pops into his Fox News feed, I'm sure we'll hear this in the next press conference.
And then he'll accuse everyone of derailing it and being against him and everything else. That's right.
I could not tell from what little I saw what adeno serotype this was. Do you have any information?
Yeah,
I think it's 2 different ones.
Yeah,
I
pipeline
think the first one is type 5 and the second one is 26.
Yes, exactly right.
Okay.
Yeah, so John Cohen had a good article in Science on this that summarizes what we know about it.
So John Cohen had a good
Prime Boost.
Has a good article, and there's also in the pipeline by Derek Lowe, where he says, they have used these vectors for Ebola vaccine candidates. And, I think Putin said, we already made an Ebola vaccine, but apparently that's not really been used that much.
Right.
So there is a nice quote here in the John Cohen article by Svetlana Zavidova, a lawyer who heads the Association of Clinical Research Organizations in Russia. It's ridiculous. I feel only shame for our country.
Oh, really?
Yes.
Is she writing that from Siberia somewhere in a gulag?
Yeah, I don't know.
So I would like to be pedantic here for just a moment.
Please.
Okay. Niles, are you listening? He reminds me almost every week that until a vaccine is, and I don't know what the right term is. Sorry, Ed. Whether it's approved or licensed. There's different terms used for vaccines and drugs. But until the FDA blesses whatever it is a vaccine candidate.
Yes, of course.
Right.
Not a vaccine.
I think we say that. I think we say that here at TWiV. Yeah, for sure. Okie dokie. That was...
the one thing I will say about Lizzie's question is that she wants to know how to convince people that they shouldn't want this vaccine. And I'm a little bit surprised that she is convincing a lot of people of that. I feel like in my little bubble, I'm hearing more questions about how do I know that a fast vaccine is safe? People are afraid that our vaccines are too fast instead of the opposite here.
I think it's very difficult to convince people, but you have to repeat our conversation, Lizzie.
And also, point out that China had one already too, that they've actually got it a little further phased than this. So why not get the Chinese vaccine instead of the Russian one?
Could apply the same logic here, Brienne, that most people do that don't speak a language, but when they want someone to understand them, they just speak louder.
No, that's a common tactic
Yes,
online.
yes.
Rich, can you take the next one, please?
Britta writes, Dear TWiV team, thank you for your excellent podcast. I'm grateful for the time you dedicate to educating your audience. I've been a loyal listener since March, and I have a TWiV notebook full of information I learned from you. My family knows not to disturb me when I'm busy TWiVing. As an amateur scientist, listening to TWiV has been a joy. Last week, August 6th, on the Political Gabfest podcast, the host David Plotz mentioned TWiV 640 during their cocktail chatter segment. He talked about Michael Mina's idea for the cheap at-home testing. This week, 8/14, Michael Mina was a guest on Political Gabfest. Michael spoke on a general public level about how cheap frequent testing might work. It was a nice segment, and they provided a link to TWiV 640 in their show notes. Thank you again for your amazing and important work. Britta in PA, where it's currently 78 Fahrenheit and rather humid.
Indeed.
Thank you, Britta.
Yeah, I didn't
know. Nice to know that TWiV is percolating through other media.
So Britta is filtering the...
yes.
Oh, sorry.
Dixon. Brienne, can you take the next one?
Sure. Gregory writes, Dear TWiV, in TWiV 648, you discussed a box and whisker plot showing CT values in 3 age groups. In a minor slip-up, the whiskers of the plot were described as being error bars. In fact, things are more complicated than that. I feel like that's just a general take-home message. The box itself is not too bad to explain. Put simply, half the data lie in the box. The whiskers are more complicated. To be honest, I don't think it's worth going into. Let's just say that the whiskers are decorative. The important point is this. Error bars reflect our uncertain knowledge of some quantity, for example, the standard error of the mean. In contrast, box and whisker plots show the variation that exists in the population. Imagine that you tested huge numbers of people. Error bars would shrink to 0 as your uncertainty decreased. In contrast, the spread between the 25th and 75th percentile will be whatever it is out there in the world, so the box plot would not shrink. Nothing like catching up on TWiV after a long day of making plots. Keep up the good work, Greg. And then Greg has a PS from the documentation of the ggplot2 software. The upper whisker extends from where the hinge, top of the box, to the largest value no further than 1.5 of the interquartile range from the hinge. Too complicated, in my opinion. People should just plot all of their data points and then report summaries, p-values, etc. And Gregory is a staff bioinformatician in New York City.
I believe that was my error, so apologies.
Well, I think we all probably did it. I think this is a lovely way to point out in a minor slip up. And I had another email from someone a few weeks after, or just after this, who said, I was horrified to hear you call them error bars. And I said, really?
Horrified.
If you're horrified at this, what is the pandemic making you feel, right? Come on. Horrified.
Here.
So, Greg, you got it. You have the right touch.
Thank you, Greg.
Have the right touch.
And I will I'll never be able to describe this. You know? That's okay.
Too complicated. He said there's better ways to do it. And I think, why not?
Good
why
point.
Not?
Right.
All right. The next one is from Tim, who writes about R0. Dear TWiVers, the 3 digits after the decimal can probably be most accurately presented as .ish.
Ish. Yeah. Good.
How much precision is lost versus the mistaken appearance of precision? Tim. It's brilliant.
I like that.
It's good.
Nice.
3.258, 3-ish.
Yeah, 3-ish. Yeah, really. Until it's like 3.5, then between 3 and 4, right? I mean,
yes.
Yeah.
It pretty much summarizes my command of statistics.
Yeah.
You know,
finding the value of ish.
Yeah, I mean, sometimes you can add a lot of.
It's got an N over it.
Sometimes you can add a lot of decimals, and the number of the amount of data you have doesn't justify it, right? I mean, so yeah, you don't want to.
I mean, that's why there is a Department of Biostatistics, because you can go to them for these kinds of things.
Yes.
And I did that all my life, and I never felt embarrassed about that at all.
No. If statistics was intuitive, we wouldn't need it.
Exactly.
Right.
Or if the data was so obvious you didn't need them, then that's that.
And that's also why we teach students about significant figures.
I have a book on my shelf. I'm trying to find it, but it's just the funniest title. I had it in grad school. It's called Intuitive Biostatistics.
Are the pages like blank or something?
The significance of insignificance.
Is it in a series with intuitive immunology?
Yeah, right. Alan, you're next.
Sure. Siobhan writes, I desperately want to get back to in-person rehearsals for my choir. I live in a relatively low prevalence area, cumulative incidence less than 100 per 100,000 of the population. And we're currently in the second wave of the pandemic with a 2% positive testing rate, standard qPCR, and seroprevalence less than 0.5% for anti-S1 and N antibodies. I'm reluctant to believe there is substantial pre- or asymptomatic spread in the population. All things considered, my choir of 24 young adults is considering having in-person rehearsals in September. We would be taking precautions such as rehearsing outdoors, weather permitting, physical distancing when indoors, self-screening for symptoms, improving the ventilation and sanitation, etc. Just for reference, 100 per 100,000, I would consider kind of middling to high-ish. My local area has a rate of 4 per 100,000, so maybe I'm a little biased, but continues, I'm only a virology master's student, but I'm well equipped to run an assay or two each week. It might not be optimized for clinical diagnosis, but I can ask my fellow choirsters to spit into a tube 3 days before rehearsal, screen them to the best of my ability, and use the results to decide whether or not to hold rehearsal. In fact, my lab is collaborating with a group that's developing an isothermal testing kit similar to the LAMP kit used by those firefighters. What do you think?
I don't think it's a good idea.
No.
I mean,
there's liability involved.
There's liability.
I don't.
Yeah.
To me, it's beyond legal liability. It's moral liability.
Yeah.
I mean, she says, Siobhan says, I'm reluctant to believe there is substantial pre or asymptomatic spread. There is. You see what happened in New Zealand? They say we have no cases, and now they have cases because they're
somebody got in. Yeah.
So I mean,
I don't think in New Zealand, I don't actually think that they had asymptomatic spread for 3 months, given their level of testing. I think somebody came in with it and they didn't immediately catch on to that, and
maybe
something slipped at the border. But 100 per 100,000, where are those 100 people getting it if there's not significant spread going on? It's circulating in your community and it's out there.
Yeah.
And I mean, I've mentioned several times my daughter was going to a tennis camp, and we had an extensive family discussion, even with our 5 per 100,000 at the time rates locally. Is this safe? It's outdoors, and they've got measures in place and everything. And that's tennis.
Yeah.
Choir is kind of the poster child for super spreader events, right? One of our earliest super spreader events was choir practice, because you're there, singing your droplets onto everybody else. This is a really potentially hazardous situation, and doing this this vigilante testing approach, winging it on your own. I understand the motivation, and I applaud the motivation, but I think you're going to feel awful if this goes pear-shaped.
Right. And Siobhan, I would also worry about whether that's going to put you at risk by spending a lot of time with these samples. What kind of precautions are you going to have to not acquire disease from the many samples you would be working with? I don't think this sounds like a good idea for you. or for your choir.
Yeah, and I know Rich properly shifted the focus to the ethical issue, but I do want to just go back for a moment to the legal issue here, that if somebody in your group gets sick and has serious symptoms or passes it on to somebody who has serious symptoms or dies, you could really be on a big legal hook here.
There's something here that I kind of don't get. Okay. Like, yeah, sure. There's lots of things I would like to do, but there's a pandemic. Okay. So just chill.
Yeah.
You know, I can chill for a year. Fortunately, as we've said many times before, I don't have to go to work. And so, you know, there are things that people have to do, but you don't have to sing. Just chill. It's okay. Be patient.
Then continuing, I've seen evidence for a chlorhexidine mouthwash reducing viral load in saliva, which presumably constitutes most of the respiratory droplets shed while singing. In Japanese dental practices, povidone iodine is similarly used to reduce viral load. That is something that could be used in this context. But again, I don't think that I don't think you can reduce the risk enough. And this is something I believe we talked about with dental practices, right? On that episode.
Yeah, Michael did talk about it. But of course, you know,
Michael talked about
the patient.
But I mean, a dental practice, they give them that. My daughter just went in to have her braces adjusted, right?
Yeah.
Then they did this whole thing, and I'm sitting there in a mask, and they're, you know, everybody's masked up except her. They had her rinse with, I think, this same thing, chlorhexidine. And then after a couple of minutes of the hygienist working the braces wire out, they had her rinse with it again.
Yeah.
Right.
So what are you going to do? Every few bars, somebody take a swig?
Yeah, I mean, the singers are going to be exhaling, and that's going to be coming from deeper in, and it's not going to be inactivated by this. So this will not help. No, not at all. Sorry. I'm very sorry. Yeah.
Sorry. Siobhan, I empathize. There's a lot of stuff I'd like to get back to doing too, but
same.
This isn't gonna.
Actually, I just want to TWiV. I'm really happy to do this.
You're happy.
Actually,
TWiV and chill.
I don't know what kind of choir Siobhan participates in, but the singing group that I participate in has taken this opportunity. We still meet weekly, Zoom. We've done a couple of singing exercises. They're really hard. But we've used the opportunity to have music theory classes, to have coaches come in and talk to us from various venues, to watch and critique videos together. Oh, and nice. In some ways, I've learned as much or more during the pandemic, and sort of maintained the social contact with people as normally. So there's things you can do to maintain that activity without actually getting together and singing.
Yeah, and you can do music lessons.
Yeah.
My daughter was doing this on the piano until the end of the school year, and she'll start up again in the fall with the same thing. And it was actually in some ways better. I mean, it was a lot. I didn't have to drive her there. We just set up the laptop and next to the piano. But singing lessons or singing, one other person coaching you on your singing, that's totally something you can do through this medium.
Dixon, you're next. Hold on.
You'll need to unmute yourself.
I muted you.
Oh.
And why isn't the unmuting working?
Because I am in control.
I muted you because you're breathing way too much, Dixon.
Ronald, by myself. Who do I control here, right? Ronald writes, thought you might like to see how our wonderful state is handling the pandemic, the state being Florida. Also thought your face needed a palm today. That means, I guess.
No, it's a palm.
It's a reference to an NBC News commentary that says there's a Florida sheriff that actually bans the use of masks by his attending police force.
I don't understand this at all. So
it's a defiance of laws by a law.
This is
it's crazy,
beyond willful ignorance.
That's right.
Well, he says the sheriff says, if you look at the experts, half of them say they work and half say they don't work. So we're not using them. I mean, if it really
use
is,
the half that work.
Yeah. So why do you ban them if half say they work? I don't get it.
Yeah, right.
And what's the problem?
Right.
Why is a sheriff? I mean, what's the legal thing that you would have to ban? I don't get this at all. At all.
These are the people who would, you ever get those little, they come in like dehydrated foods and other types of containers, a little package, the desiccant says do not eat.
Yeah.
These are the people who would say, F you!
That's right. No.
It's just oppositional. It's just somebody said I shouldn't do this, or somebody said I should do this, so I'm not.
That's right. That's right. They're contrarians.
Yeah.
That's exactly
so that's
what
entirely. I do understand what it is, but it's infantile.
Yeah. This is
anybody who's
how did anybody become a sheriff?
Seen this.
Well, yeah,
that mentality.
They got elected.
They got elected.
Dixon.
Yes.
Is that what it is?
With that mentality.
Florida. Come on, you live there. Have you encountered people like that, Rich, in your lifetime in Florida?
Well, actually, I live in a bubble. Okay. And always have. Okay. So my direct encounter with this sort of mentality is pretty rare, but it's there.
Rich, can you take Anthony's, please?
Anthony writes, like language, music, and math, or should that be language in all its forms? From a small set of rules and a limited set of building blocks, viruses construct an unlimited number of unique statements. With virology and infinity, I think that there's more to it than that. In the mid-70s, a Jesuit instructor in high school theology class explained that this was the greatest subject. With science, at some point in the distant future, everything will be known, but since God is infinite, something always can be learned. Focusing just on his comment about science 45 years ago, that seemed reasonable. That it was humanly possible to unravel the thread, even though that would take a long time. Now, my impression is that every answer uncovered about the nature of reality brings with it more questions. So, like the priest's approaching half a century old reflection on theology, virology is infinite. For what it's worth, that's Anthony.
I like it.
Yeah,
very good.
I like this. And I did a couple of years ago, I did a sort of a weird essay on my blog about this concept of the infinity of science and the infinity of biology in particular, and that this resonates.
Okay.
And I find it, appropriate that it is a Jesuit instructor who says that since God is infinite, something can always be learned. Because that's not your sort of standard theology.
Aren't the Jesuits into academics, right?
The Jesuits are an interesting group.
Yes, they are an interesting group.
Yes.
They're an intellectual group.
Yes.
Yeah, like they are the ones who run colleges often. Right?
They're the brains behind the operation in a lot of the Catholic. Yeah.
Okay.
Not all of them. But,
not all of them.
Now, my friend Bill Tramontano, he's listening, and I think he's told me that Tony Fauci went to a Jesuit school. Holy Cross, right? Holy Cross. Okay. Correct me, Bill, if I'm wrong.
I think that's right.
Brianne, please.
Jenny writes, "Hi, TWiV crew. I would just like to draw your attention to a past TWiV episode from the 18th of March with epidemiologist Stephen Morse, who at 44 minutes and 25 seconds discussed rapid at-home tests for SARS-CoV-2, even if not completely reliable. He lamented the fact that there didn't seem to be the motivation for them and indicated how and why they would be of help. I can't believe that was almost 5 months ago now. I was super impressed with the Michael Mina episode, but I can't help wondering how things would be if this was a thing back in March. I have been listening for the past 100 or so episodes, many multiple times, and currently burning through the back catalog. I have always loved science, and I'm glad I discovered you. Thanks for all you guys do, Jenny. And Jenny is a registered nurse in Australia."
This is brilliant, Jenny, that you remembered this. You picked this up.
Yes.
I forgot about this, but he did.
Yeah, me too.
Yeah.
He did say. And then when I said to Steve, I had a meeting with him, and a Zoom meeting, I said, "Oh, Michael Mina." And he goes, Well, I'm glad to see I was right.
Thank you for paying attention, Jenny.
You're paying attention. Really good job, Jenny. That's great. All right. Michelle writes, hi, Twivers. I've been following since around February, and this is one of my thanks to you. Encapsulated ignorance spreads through the web. TWiV inoculates. That would be a haiku, right?
That would be a haiku.
Yes, that would.
Stay safe, stay grumpy. Michelle, a breezy 79F in San Jose. That's wonderful. Love it. Alan.
Maxim has a question. Can rain kill viruses? Right now, my town is disinfecting the streets every day, twice against COVID-19. However, the weather over here in eastern Spain has been quite rainy in the last couple of days. I wonder if rain can also be an effective natural disinfectant. 2 weeks ago, the country entered a state of alarm. You cannot go out except for work, medicine, or groceries. Police and military are patrolling the streets and roads all across the country. And if they find you in violation of the curfew, you'll be fined between $700 and $25,000. If you happen to be a government worker, which also includes teachers in public universities, you can even go to jail. So far, only in my region, more than 5,000 people have been fined. You could think of our situation in regard to the coronavirus like a lighter Italy. I think this is from...
Must be from a while
ago, right?
Yeah.
Okay. We're finally getting into our backlog. Great.
Indeed.
Healthcare in Spain is decentralized, although we do have public national healthcare system. Each region is free to manage it however it wants. I'm more afraid for you Americans since your national and regional governments have been rather passive to this threat, and I'm afraid many people are going to die over there because of your privatized health system and a more individualistic society. This is from a few months ago, and it is rather prescient.
Yep.
Yes.
I think
rain is not really going to be a disinfectant, but on the other side of things, whatever they're spraying the streets with probably isn't going to help much either. So
I think part of our problem here is also we haven't had leadership.
Yes, that's a big part of our problem.
Yeah. We got a pretty scattershot healthcare infrastructure and no leadership to.
Yes, an expensive but scattershot healthcare infrastructure. So this rain will remove stuff from the streets, but you're not catching SARS-CoV-2 from the streets, which you've probably already heard us discuss on TWiV if you're still listening to the show. This is a respiratory virus. You get it primarily by having people exhale droplets into your face and close contact and such.
Right. These older letters are going to be an interesting reflection on how far we've come and how much we've learned.
Yes.
Dixon, can you take the next?
I can. Edward writes, hi, Team TWiV. In episode 594, Alan said to avoid sprouts. With brassicas being among the most beneficial, healthy of vegetables, can I ask Alan to qualify his reasonings, please? With regards, Edward. Okay, go ahead.
Food poisoning. If you talk to people, I've covered conferences on food safety and talked to a lot of people who work on food safety, and every single person in food safety, if you ask them, is there any food that you just that you won't eat ever? Like just you don't care, you're not eating it at all. They will all say sprouts.
Yep.
Wow.
So what is it about sprouts?
What it is about sprouts, and specifically raw sprouts? I want to point that out. So you take seeds, which came off plants, right? Somewhere growing in a field with maybe some manure and dirt and everything, and you put them in water and you let them sit. And it's totally non-sterile. You just threw dirt in water and then you're going to incubate it at more or less room temperature for a matter of a few days. And it's this nice, moist environment and everything's just growing all fine. And you're going to take that with no kill step of any kind. There's no heating of it. You just take it out and eat it. Yum. This is not just a conceptual thing. Sprouts have been the source of some of the worst foodborne disease outbreaks ever, including the worst E. coli, enteropathogenic E. coli outbreak ever, which was came from organic, locally grown sprouts. sold at a farmer's market in Germany, which, in case you hadn't noticed, is not an under-regulated country. So this should have been like the cleanest, healthiest thing in the world, and it caused a huge E. coli.
Do they not at least rinse these things or something?
They could rinse them, but it's not going to do anything because they've got all these little. I mean, it's a little baby plant, and it's got all this surface area curled up. And
no, the seeds. I think Alan meant maybe or rather Rich meant the seeds.
No, I meant the product itself.
Yeah,
but I think of it as
it's impossible to rinse off.
No, it's
high surface to volume ratio.
It's something that is particularly easily contaminated at the beginning when you're getting those seeds, and then is not washed.
And even if it's washed at the end, it is impossible to get fully clean. It's not like strawberries or something where you hose them under the faucet and you've just cleared the dirt off. They're just not cleanable.
Well, fortunately for me, I don't have a real overwhelming craving for sprouts anyway.
No, I'm not that into sprouts either. But if you're into sprouts, cook them. Raw sprouts. Don't do that.
I stopped eating sprouts a year after that outbreak in Germany, and I learned at an ASM meeting everything Alan just said, and it's not worth the risk. Yeah. Rich, you're next.
David writes, I'm a junior doctor in the UK. I wondered if you think it's possible that the mode of transmission could influence the clinical manifestation of the disease. For example, if a person is inoculated via aerosol, would they be more likely to develop a lower respiratory tract infection due to the deeper penetration of the smaller particle size into the lungs? I would say yes in this case, but we can debate that if you want, because I have heard that particle size can influence where in the lung the virus particles track, and that could influence the pathogenesis of the disease. You want to argue with me on that one?
Well, I think everything you said is correct, but I think that if you were to take this and say, well, what about mode of transmission where you ate something that was contaminated? Would you then get GI symptoms? I wouldn't be able to take this that far.
No.
I think place in the lung, like you were just saying, makes perfect sense, but I wouldn't take this any further than that.
So, Brian, I'm sorry. I was going to say that New Zealand has 4 new cases, and they're blaming it on some imported chicken that they made chicken wings with. So I wouldn't be too sure that you couldn't catch it this way.
No, I'm not arguing that you couldn't catch it that way. I'm arguing that you wouldn't have a different disease.
Oh, right.
It's not that you would have suddenly some sort
disease.
Of GI SARS, there's GI COVID.
But there is ACE2 receptors in the GI tract. So
right. Yeah, but I don't think, Dixon, that you get initiation of infection in the GI tract. I think it's post-respiratory infection. You swallow virus. You swallow virus and it reproduces maybe in your tract, and then you shed it. I don't think you initiate infection.
So the chicken they ate, they inhaled their food?
Maybe when you eat, you inhale, right? Chewing generates an aerosol.
Yeah, and
that's preparation.
That's a theory. I mean, New Zealand right now is kind of scouring the possibilities of how did this get in. My money is on somebody sneaking past something. Yeah,
I'm sure,
but it's in one family only.
I don't think it was the chicken. I think there's a person somewhere who didn't get tested properly or didn't quarantine properly, or something happened at a border and somebody got in and brought the virus in. To this question of different disease with different infection, I'm not sure I buy that for SARS-CoV-2. Just because we don't have the evidence of it happening that way, and we have this kind of clean demographic breakdown where it seems to be more about age and other factors and comorbidities, and it's not so much about where you were exposed. This could be going on, but I don't think it's a big deal. I do know that this is a big deal for other viruses.
Okay.
And in the case of avian flu, we've talked about this if people go back to the earlier episodes, where you've got avian flu strains that nobody outside of poultry workers gets. But poultry workers who are exposed to large quantities of chicken stuff in aerosolized format all day, it gets deep enough into the lungs that it cannot get access to the right receptor, and that causes the infection, but it doesn't spread to somebody else. So these are these one-shot spillover events that we get where the deep lung infection is different from the shallow lung infection.
Yeah, totally agree. Yeah, I agree.
I do want to use this as a small teaching moment, because in my experience, I have 2 examples of where route of infection affects the disease outcome. One is in the case of variolation, the original practice for smallpox immunization, which involved taking material from a smallpox pustule and scratching it into a person's arm, which or smallpox was ordinarily transmitted as a respiratory infection, but if you transmit it artificially by scratching it into your skin, the mortality is reduced from 30% to about 1%. Okay, I think because you're skipping or mitigating the systemic spread of the virus that you ordinarily get through a respiratory route. And the other is one we've talked about before, which is the adenovirus vaccine that's used in the military, which is a wild-type adenovirus that's ordinarily transmitted as a respiratory infection. And the vaccine is that same wild-type virus that's encapsulated in an enteric-coated capsule. And you swallow the capsule, and it stays intact until it gets into your intestine, where it dissolves, and you get an infection of your intestinal tract, which causes no symptoms, but does cause an infection and you mount an immunity. Okay, so it is clear that the route of infection can, although those are 2 artificial circumstances, can influence the outcome of an infection, the pathogenesis.
Rich, you're next. I'm sorry, Brienne is next, not Rich. Brienne.
All right. Maurice writes, my doctor's son is a radiologist working in France, Maubeuge. He recently forwarded me his CAT scan of a confirmed COVID-19 patient showing a faint ground glass shadow in the lung. He assures me this would not show up in a standard X-ray. I say this because Alan Dove in TWiV 594 mentioned that his wife with signs of pneumonia was sent home from hospital for self-isolation because of a negative X-ray. Beware.
Yes.
Hey, and the part we didn't highlight shows the need for quick COVID tests as a system.
Yes.
Wow.
And as a long delayed follow up to that, she has subsequently tested negative. Yeah. She has not had an antibody test, but she got a SARS-CoV-2 test about a month ago and didn't have it then. And we kind of think that the pneumonia symptoms she had, since they passed so fast, and it was probably not it. But it was interesting that it was handled in that way. And yeah, at the time, I thought that was a little sketchy, that all they did was a chest x-ray and say, you just go home. Yeah.
Cornelis writes, hi, TWiV team. A little update from Germany. Our CDC-like Robert Koch Institute, RKI, does provide detailed statistics about cases in Germany. The numbers differ from CSSE because of different... CSSE is the Hopkins-sourced stuff, because of different algorithms and also how cases are submitted, and provides a link to that. You can either look at German states, Bundesländer, or focus on regions, cities, Landkreis. Also, there's an overview on different maps and provides another link, but as far as I can see, only in German. Stay safe and healthy. Thank you, Cornelius.
Cool.
Very good. Alan Dove?
Sure. Visto writes, I've seen reports there are 2 strains of the SARS-CoV-2 virus, the first of which is 98% similar to a bat coronavirus. So that seems like the inspirational source. The second strain had 29 nucleotide region deleted from an important open reading frame that splits the ORF in 2 and reduces the replication ability of this virus. It's been reported the strain with the ORF8 segment into ORF8A and ORF8B shows reduced severity of disease. Original intact strain caused greater death and so forth. Could not the attenuated virus be a proverbial cowpox to the smallpox that is the intact SARS-2 virus? I would like your expertise on this. So this Vincent loves to talk about this. He loves to talk about the mutant SARS-CoV-2, right?
All right. So you're doing a little confusing here, Visto. So there is, with the original SARS, there was a deletion that split ORF8 into A and B, and it didn't show much reduced severity of disease in humans. It just predominated after about halfway through the outbreak of SARS-1, and one of the hypotheses is that it might have reduced virulence and allowed patients to live longer and transmit, but that's completely hypothetical, and there's no evidence to support it. That's SARS-1. Now, not too long ago in Singapore...
And SARS-1, by the way, killed a bunch of people and was a highly lethal virus.
Yes.
Indeed.
SARS-1 is very different from SARS-2 because over half of the infections, you went to the hospital. And so it was very easy to contain it because you were in the hospital. There was very little community spread, very little asymptomatic spread as there is with SARS-2. All right. So now, not too long ago in Singapore, viruses begin to circulate, SARS-CoV-2 viruses, with a deletion of ORF8. We don't know what that means for the pathogenesis whatsoever. So to suggest that it might be, like cowpox and smallpox is not really, we're not there yet. We can't say that. It might be that deletion of ORF8 does something interesting, but we don't know that. And Amy in my lab is working on that, so stay tuned.
I'm glad to have that story refreshed because I had forgotten that. Now, what's the prevalence of that deleted virus?
It's very low. It's very low. It's just been reported in Singapore, and I think Taiwan. Amy's going to yell at me on Monday if I get it wrong, but sorry, I just don't remember right now.
Don't go into work.
I don't on Monday, but still there's this thing called the internet where you can yell at me.
I wouldn't answer any of the email.
ORF8 is very interesting. And so actually, let me just say a few more words. So I heard Christian Drosten, he's the guy who worked on the SARS-1 ORF8 story, and I heard him in Rotterdam at European Society for Virology meeting last year talk about this, and I had never heard about it. And so I got interested. I went back and I read the paper, and then, boom, SARS-CoV-2 emerges in January, and I emailed him and I said, does it have ORF8? And he said, so far it does. I said, because that's interesting. And then a few months later, it started to be deleted in SARS-CoV-2 as well. So it's really interesting, but the significance is not known whatsoever.
And nobody knows what ORF8 does, right?
It gets deleted.
There are some I'm not going to say anything. There's some very interesting suggestions about what ORF8 does, and it's being worked on. Yes, it could be very interesting. Okay. Who read that? Brianne? No,
I read.
I did not.
I think I read
so
that.
Dixon, you're next.
Yes, Emily writes, hello, TWiV. I am a structural biologist. So my question is this. How do we know which protein to make for serology tests to best approximate what's on the surface of the virus, and so what the immune system has seen or would see for vaccinations? Making the wrong approximation may cause serology tests to miss antibodies or to misrepresent which antibodies bind the actual spike protein. Take it, Vincent.
It's a great question. And that's,
yes, it is.
This is why we make pre-fusion forms of the spike, which prevent it from undergoing the conformational change, which wouldn't be representative of what you see. Yeah. So the vaccines all have pre-fusion, and I'll bet the ELISAs do as well, right? What do you think?
Let me ask you this, because I did serologies a long time ago for a different organism, of course. But the way I approached it was to take a number of infected animals, because this is a zoonotic disease called trichinosis, and we pooled their serum, extracted the IgG from it, and then attached it to a Cephalus 4B column and used it as an immunoabsorbent surface. We percolated a crude mixture of the worm down through the column, and the only thing that stuck were the antigens. And the antigens that stuck were the only ones that the pigs responded to. So when we washed off the antigens and got a solution of that, it was the same for people, by the way. They both, pigs and people have a lot in common, not the least of which is their immune system. And the next thing you know, we were clarifying all the false positives and false negatives. They all fell out, and the only thing we had now were true positives and true negatives. So why can't you do that with the virus?
Exactly. So basically what you're trying to do is identify what I would call the immunodominant epitopes.
Yeah, exactly.
Nice.
Okay.
Sure.
The things that show up most frequently, and use that for your diagnostic. And I would hope somebody's doing that somewhere.
Yeah, but I
mean,
think
you could use whole viral particles for this, couldn't you?
Well, no, then you'd have to inactivate them, and that might, because you don't want to have to work in a BSL-3, right? So you'd have to inactivate them, and that may change.
What about a hybrid virus where the internal components?
You could use a pseudotype, and people do use pseudotype VSV
pseudotypes
for their serological assays. Remember?
Sure.
What was her name?
Right. The one that did
Viviana Simon. They used pseudotypes in parts. So does the Rockefeller group. But other groups use protein. And I don't know. Do you think, Brianne, they're using prefusion versions?
I think they're using prefusion. I think that that idea has been in the literature for a while with some of the, say, SARS-1 and some of the other coronavirus spike proteins. And so I think it was translated pretty early on.
So Western blots probably would only be partially informative, right? Because you miss all the conformational epitopes.
Exactly.
Yeah, we actually, so a long time ago, we did the paper out of Rockefeller Group where they looked at sera using pseudotypes and infectious virus, and we asked, did you do Western blots? And Theodora responded to me, we didn't see anything in Western blots. Because that's very common for HIV analysis, right? You do Western blots and you pick it up. In fact, that was an early antigen test, right? A Western blot.
It was.
Yeah, that's right. That's right.
For HIV.
But these are things that would be really nice to know because, always in the back of my mind, and as we've discussed all along, what other antigens other than spike or what other regions on spike might be relevant epitopes for immunity.
Well, so you need an animal model that really imitates this infection, and I guess the rhesus monkey comes close, but
no,
it's pretty expensive research.
It doesn't. There's no serious disease in rhesus in any non-human primate. There's no serious disease in any animal model so far.
Well, I don't mean the disease. You could prevent the infection if the vaccination actually worked.
But you said mimics the disease, so I'm being picky.
Well, I did say that. You're right. Sorry, I misspoke.
The nuclear protein is also important. We know that antibodies against N will also neutralize infectivity. So that's another important one to measure. And I think we had an email earlier that talked about that. But yes, these are interesting questions for sure. Maybe we need to get someone on who works on this.
It's a very good question.
Yes.
We need to get someone on who works on this. Okay, Rich, you're next.
Michael writes, we use several food-safe sanitizers for fresh produce and surfaces. Much better than bleach, soap, and rinse. That disgusted Vincent so much that he gave up on lettuce. And he mentioned several of these: chlorine dioxide, hydrogen peroxide, UV, corona discharge ozone.
What a great name, right?
Yes.
A little help from your friends. We make chlorine dioxide solution on farms for fresh produce wash. Here's how we do it. But first, a few warnings and details. Chlorine dioxide is unstable and will spontaneously combust. And he gives a link to that. The short and the please don't try this at home answer is interesting. Even with all those dangers, if used in dilution, chlorine dioxide is highly effective in surface sanitization of fresh produce without bad taste or destruction of tender things like lettuce, and makes a wonderfully effective produce wash with no rinsing required. The corona discharge ozone is my current go-to for room and cloth item sanitization after laundering. We apply it in nightly unoccupied areas for about 2 hours on a timer or in a chamber or cabinet for masks and small items. I would think this ozone would be good for N95 masks. I wonder if NIH is trying it. P.S. Careful with microwaving N95 masks containing metal bits like strap attachment and bendable nose clips. Those metal bits become RF antennae and will create some exciting fireworks and possibly fire in your home microwave. And that's from Michael, who is at Planet Earth Diversified.
Cool.
So this corona discharge ozone.
Yeah, it makes ozone. We talked about ozone a while ago.
Yeah, and we highlighted this letter. It's a long and very, very informative letter. So those were some excerpts of it. I didn't think we needed to spell out the procedure that produces the chlorine dioxide.
No, but he's got links to all of this stuff.
He's got links to all this stuff, and the corona discharge ozone comes from a $200 device. But sounds like you don't want to be in the room.
Early in the outbreak, we were wondering if food would be a problem, but I think we have found that it really is not an issue.
No.
Yeah.
However, I've resumed eating lettuce because it's from our garden, and that's fine. That works. Although now we're past no sprouts. We're past the lettuce season. It's too hot now.
Yeah.
Brienne.
Lisa writes, is it transmissible before it is detectable in the current tests? Of course, meeting SARS-CoV-2. And Lisa is from Georgia. I would say yes, Lisa, depending on which current test we are talking about.
Who is it?
Yes.
These are letters from a while ago.
Yes.
That's right.
Yeah, I mean, that's the whole key, that PCR will pick it up even before it's transmissible, although the rapid antigen test, the Mina tests will not. But we really only want to know when you're transmitting, right?
Yeah. Right. And certainly if you had an antibody test, that would also only be detecting things relatively late.
Yeah. Our next one is from T, the letter T.
Right. This episode brought to you by.
That's right.
Is that Sesame Street or something? Right. So this is a very long.
Yes, that's right.
It's a very long, well-referenced letter about masks of different sorts.
Yeah.
Which we talked about a while ago, and just a few excerpts here. I would like to suggest homemade masks as a guilt-free alternative. PPE to be used at times when strict physical distancing cannot be adhered to, e.g. grocery shopping. Provides 3 peer-reviewed articles on the efficacies of homemade masks.
Yes.
And you could take a look at those. However, it should be noted that the studies cited above were done with previously unworn masks, while washing and drying has been shown to lower the filtering efficiency of cloth masks. And he gives a graph showing filtration efficiency of different test materials and so forth, how to wear them, a nice diagram in multiple languages. And T is in Hesse, Germany. And you can tell when this was sent because it's 8 degrees Celsius.
Right.
Probably about March, I would guess, right?
Yeah. So that's when we were first talking about improvised masks. And now, of course, we all have stacks of masks that we can improvise to go into the grocery store.
So there's a new study out, I'm sure you're all aware of this, by the engineers at Duke that took about 8 different commercial masks and neck gaiters and compared them, and
this has gotten huge amounts of hype. And the reason
so, what do you think about it? What do you think about the neck gaiter?
Okay, so these
Vincent
studies,
is giving it the thumbs down.
All right, yeah, let's talk about mask studies, and we're talking about the improvised little strips of cloth people are putting on, not the properly fitted N95s. So
right,
what we don't have and what we're not going to have for these improvised masks is really good infection epidemiology data proving like what percentage efficacy you get from putting a bandana over your face. And the reason we're not going to get that is because you can't ethically do that study where you randomize people to a cloth mask and a no mask group and you have them all sing the parachute chorus.
It's the parachute control.
It's the parachute thing, right? We've got enough epidemiology evidence that these probably do some good, and they cost nothing. And there's no reason not to. So wear your mask. All right, that's first thing. Second thing is, because we can't do those studies, what we get instead are these laser light show studies. right? Where somebody, we've
right.
cited some of these where you've set up a green laser and you have somebody breathe and you see the droplets coming out, it visualizes it.
Mhm.
These get massive news coverage because there's video and pretty lights. and I'm not saying we shouldn't do those studies, and I'm not impugning the researchers who are doing them. There's some good work being done there and analyzing the physics of breath, and we're getting a lot of good data. That's great. But we can't really put a lot of stock in what we get out of those in practical terms, because it's not measuring infectious virus. It's not even measuring necessarily droplets that could contain infectious virus, right? So this thing that just came out that neck gaiters are evil, which has gotten all the coverage, because a lot of people, including me, I've got a bandana. I've also got a neck gaiter. It's very convenient. You flip it up over your face. And a lot of people have been using these, and now people are going around saying, oh gosh, my neck gaiter doesn't work. It's probably fine. What they found was that in the particular neck gaiter they tested, when you say the phrase "stay healthy" into a green laser, it apparently doesn't stop the breath completely. It allows some to come through. So they're saying that the droplets get broken up into smaller, sort of wafty aerosol that might contain infectious particle. But again, we're not looking at infectious virus, and it just, I think it confuses an issue that people really don't need to be confused on right now.
I don't think we needed that study, which, as you said,
what you've got.
As you said, they didn't look at virus, and that's all I care about. I don't care about droplets glowing in the laser light. It doesn't
and
matter.
the other thing you said is they found this with the particular neck gaiter that they tested. They didn't look at multiple neck gaiters. They didn't look at how well your neck gaiter fits, or if it's gaping not very close to your face. So you could imagine maybe if the neck gaiter they tested was loose and the masks they tested were tighter, that could have given them some of the differences that they were seeing.
Alan, you're next.
Okay. We're up to
Jessica.
Jessica wants to know whether the virus could survive and infect someone through a dog's fur. Our 2-year-old rescue pup is going stir crazy inside. I have had a head cold. My husband is working from home very intensely, so no long walks at the moment. And currently there's still the option of sending him to daycare, but we don't understand the potential risks.
Husband or the dog?
Well, hopefully, Jessica, you've walked your dog by now, but it's really unlikely that you're going to get it from dog fur, if anybody's still wondering about this. We do now have some reports of a few dogs and certainly cats, including some big cats at the Bronx Zoo, catching SARS-CoV-2. We don't think most people are going to catch it from their tigers, with the exception of perhaps some reality TV stars. So I wouldn't worry too much about the dog, and hopefully the doggy daycare has reopened at this point with some kind of distancing procedures. And
I think, as you would say, Alan, this is a low risk.
This is a very low risk thing.
Okay.
Yes.
Dixon.
And as I recall, even the laboratory experiments, dogs don't infect well or at all, and certainly don't transmit. So doggy daycare is not going to amplify virus among dogs or anything. Worst possible case scenario is your dog's fur gets contaminated by a daycare worker and it gets trafficked home. But the likelihood that that's going to carry enough infectious virus to you over time and you're going to get it is like very, very slim.
And I probably ought to go ahead and get going.
Oh, it's 3:30. Thank you.
It's 3:30,
Alan,
yes.
Alan Dove, turbidplaque.com. Alan Dove on Twitter. Thank you.
Thank you. It's always a pleasure.
See you next
Have
week.
a good weekend, Alan.
Good to see you, Alan.
Take care. Good to see you.
Dixon.
Yes, sir.
You are next.
Joan writes, I have been wondering if beards and long hair could be a mode of transmission for viral particles. It used to be that nurses, if they had long hair, had to tie it up in a bun. I am not sure about doctors with beards, but I know they told them to get rid of ties in more recent years. That's true. We've gone more informal. This is all true. So, okay, good question. Anybody have an opinion?
If CDC initially said if you have a beard and you're going to wear a face mask, you should shave it to make the face masks more effective. So
yeah. So I've always been told that when you're wearing a mask, you do want to have your hair tied back, largely because you don't want your hair touching different parts of the mask, getting under the mask and over the mask, things like that. And I know that ties have been implicated in transmission of different microbes from physicians who've had their tie potentially touching one patient and then another. So I know that has been implicated, certainly. You all can say more about tie washing than I can. But I'd always heard about tying back long hair, but that I don't have evidence on that.
So Daniel Griffin solves the problem by wearing bow ties only.
Exactly.
Yeah.
And being bald.
And you may all not know this, but he ties them all himself. These are not clip-ons. These are real bow ties.
Really?
And he sits there, and in 5 seconds, he ties the most magnificent bow tie I've ever seen. And I said, how the heck did you do that? And he says, practice. He's right. Just keep practicing.
By the way, I meant to say when Alan was talking about face masks, healthcare workers wear face masks and they protect them. So that's pretty good evidence. But Daniel also wears a lot of other things. He wears a face mask. He wears a gaiter. He wears a head covering. And, a lot of stuff.
Yeah, he looks like he's...
And I, as you see, did not shave my beard. I wear a face mask.
Me neither.
But I really have minimal contact with humanity these days.
It's why he's called Grumpy, everybody.
Sitting on Zoom is one of the only times I have my hair down.
Really?
Yes.
I mean, physically not
Rich, you were going to say something? I interrupted you. Rich, were you going to say something?
Yeah.
Okay, you take the next one, please.
Lewis writes, I'm the guy who did the big study of science podcasts, which was a pick of the week back on TWiV 500, July 2018. A recurring theme in your listener emails is how much listeners trust information from TWiV and how it was prepared to them to combat disinformation about COVID-19 that is prevalent in social media. It has long been asserted in media studies research that radio and podcasts are perceived by the public to be particularly trustworthy. Audio is a blind medium, allowing listeners to engage with the content in a more active manner. For example, by imagining the hosts projecting assumptions onto them and thereby relating to them more. Isn't that interesting? Overall, the podcast medium and the format of TWiV seems to be quite the recipe for instilling trust in science. I will keep sharing each new episode of TWiV and keep telling everyone to wash their damn hands. And Dr. Lewis is a postdoc at in Durham University Chemistry Department in the UK. Thank you.
And he contrasts it to TV, where people have less trust. And I think part of it, for TWiV, is anyway that we're honest and we admit mistakes, and that's unusual on TV, right? Does anyone admit mistakes on TV?
Right. Well, I also like that he notes that TV is often very stage managed, and people listen to podcasts in personal settings, and podcasts have a conversational tone, and they can feel like part of the conversation. So I would say that even now that people can see us on Zoom and see our video, they can still tell we're not particularly stage managed.
Or at all.
Right, exactly. So I think we're still maybe getting those benefits of the audio blind medium, even now that you can see us.
I think that one of the nicest lessons that we can project is this idea that science is an interactive and very dynamic discipline, with us, arguing with each other and saying, discussing stuff with each other.
Yeah, we can argue sometimes.
Admitting our mistakes and asking whether we're right and all that kind of stuff. It's good.
Yes.
Let's see. That was Rich. So Brianne is next.
All right. Paul writes, it looks like Paul knows Rich, and he says, hi, Rich.
Oh, Yeah. Paul. Hi, Paul. Jeez, this was buried. Are you still out there?
Apparently.
And Paul says, a prior PhD student of mine who happens to be from China and now lives in Boston, Massachusetts, told me that friends of his from China are mailing him anti-SARS-CoV-2 IgG/IgM test strips. Is there any effort to get these tests from China and test them out? Paul, that's a great idea, Paul. I hope so. And again, this is a long time ago, and yet the idea was out there percolating.
Well, it's interesting that the student is in Boston, and that's where Michael Mina is. So I wonder if they collided in some way, and that's how Michael has been working on. Very interesting. Anyway, do you know Paul, right? He was at University of Buffalo.
Paul, I can confess here. I can out him. This is Paul Golnick, who's a professor in the Department of Biological Sciences at UB. And actually, he did a couple of sabbaticals in my lab where we met at, I guess we met at a transcription meeting. Yeah, because he was a postdoc with Charlie Yanofsky and worked. He's the guy who solved the structure of TRAP. Mm-hmm. Okay, in the histidine biosynthesis. Yeah. And so Paul's worked on Vaccinia for a while. Good guy. Right. Hi, Paul.
Very cool. Ann writes, is there any benefit against the coronavirus or any other infections from getting the annual flu vaccine? Does the flu vaccine ramp up the immune system in any way helpful against other viruses? I would say it's very helpful to get the flu vaccine every year, including this one, so you don't get flu, because if you then got flu and COVID-19, boy, you'd be in trouble.
Yeah.
Indeed.
Yeah, flu might knock down your immune system. And so getting the flu vaccine would definitely be helpful in that way.
There have been various reports, I think, ultimately not well substantiated, that immunostimulants of one sort or another, BCG, etc., might give somebody a temporary, at least, resistance to SARS-CoV-2, but I don't think any of that stuff is really.
In fact, I asked
gotten a lot of
one
mileage.
of the papers actually just got published this week about BCG.
Oh, okay.
That made it look somewhat promising.
Really?
I asked Kostya Chumakov months ago about this, because he wants to give people OPV for a couple of months, general protection. And I asked, would the infectious flu mist vaccine work? And he said, probably. Any attenuated virus vaccine might give you a general boost. So if you can do flu mist, but the inactivated vaccines won't do that. Dixon, can you take Paul's?
I can. Paul writes, hi, TWiV team. These podcasts are among the world's greatest treasures. Thank you all. Are there any reported cases of coronavirus co-infection with flu? 33F and rainy in Indianapolis, but the forecasts are looking good. That was then. This is now.
Wow, 33 Fahrenheit. That was a while ago.
That's a blast from the past.
I don't know, but right now there's not much influenza at all, so we're not having co-infections. But I would
Rich,
bet.
I wanted to ask you,
Dixon. I'm talking.
Oh, I thought you were finished.
No.
Sorry.
Well, if I talk, how can I be finished?
You see how we get along so well.
Sometimes I think you don't hear, actually, Dixon.
That might be true.
Are your earphones working? Hello?
What?
Hello?
What'd you say?
I think there have been co-infections. I think Daniel noted this some time ago. Okay. That's all I wanted to say, Dixon.
Well,
now, may I have permission to speak, sir?
You don't need permission.
Rich, you're sitting in a hotbed, and I mean it literally, of temperatures over 100 degrees. And Texas is one of those great states where West Nile virus always rears its ugly head, mostly because of migrant workers that come in to harvest the crops that you're not going to have this year because of the droughts and because of COVID-19. So, but what is your opinion with regards to an infection so generalized as West Nile? West Nile is everywhere. In 1 year, we had like 3,000 cases. For every case, there's 200 inapparent infections that don't exhibit symptoms. So that we must have herd immunity to come to West Nile virus almost.
Yeah, I don't know what the story is with West Nile.
No, there's no.
Do you have any outbreaks yet in Texas?
When there have been people have. There has been some West Nile in the area. I don't know what the circumstances were, whether those were actually infections in humans or trapped mosquitoes or what, or birds or
horses even, because they do a lot of horse
it's not
too.
It's not something that is so prevalent that it's showing up on the radar.
Okay.
I don't think we have. We go ahead, Brianne.
I was just going to point out that one thing that's nice about these two this letter and the previous letter, even though they may feel a little out of date because it was not flu season right now. Right now, the new flu vaccine for this year is just coming out. I've seen some signs and had people asking me about getting a flu vaccine. So hopefully this information about co-infection and those types of things will remind you to get your flu vaccine for this year.
What's the mix of H&Ns?
I am not sure off the top of my head.
You want to know the mix? CDC flu vaccine 2020. I'll pull it right up here.
There you go.
Are you ready?
Yep.
I'm breathless.
I'm at the edge of my seat. Of course, I have been the whole podcast, but that's another story.
A Guangdong 2019 H1N1. A Hong Kong H3N2. That's 2019 also. B Washington 2019. Those are the trivalent, and the quadrivalent has A Hawaii 2019 for the H1N1, A Hong Kong for 2019 for the H3N2, B Washington 2019, and B Phuket 2013. Two different B lineages.
So are those, these are all H1N1?
No, there's a H1N1, there's an H3N2, and there's a B. There's one or two Bs depending on if it's trivalent or quadrivalent.
So are any of those driftages from the pandemic strain, the 2009?
Yeah, they are, because this is a 2019 isolate for the H1N1. It's a 2019 for the H3N2. It's a 2019 for one of the B. Yeah, so they're drifted. They are drifted. Now,
thank you.
Who's next? Now I've lost track. So Indianapolis, Greatest Treasures. Who read that? Was that you, Dixon?
I read that.
So then Rich is next. Craig, we're down to Craig. We can finish this, guys. We're almost done.
There you go.
No kidding.
I'm not going to tell you there's 1,000 more where this came from, but
right. Craig White's Dear TWiV, big fan, will likely continue to listen long after COVID-19 is gone. I am a meteorologist from Florida. We ought to have him as a guest on the podcast.
Absolutely.
Where we frequently deal with hurricane threats. To help understand how to prepare, we often use analogs of previous storms and their impacts. My question, are there analogs for the present coronavirus, say original SARS, that we can apply that in a predictive fashion help prepare and respond effectively to this virus? Thanks in advance, Craig Setzer. And he gives he's at a TV station in Miami. So, as I think we discussed earlier in this pandemic, the virus and the pandemic have characteristics that are unique to this particular virus. So we can contrast it with previous epidemics and pandemics, but this is unique to itself. We can learn from it and try and look for certain characteristics in future pandemics. But what was who was our guest earlier this week?
Adam
Kacharsky. What was his thing? If you've seen, it wasn't his thing, but he quoted
if
for
you've seen
somebody
one
else.
pandemic. You've seen one pandemic.
Right. That's right.
That was good. Now, the one thing I will say, Craig, is that we learned after SARS-1 that bats harbor SARS-like coronaviruses that can infect humans, and we should have been ready based on that, and we weren't. And that's why we're in this position.
Plus, there's a lot of, I think, there's a lot of public health type pandemic preparedness that can be done that would mitigate almost any pandemic that came along, regardless of its characteristics.
Brianne, you're next.
Yes, and Craig, I hope you enjoy our discussions of the weather. Evan writes, "Hi, TWiV. I have found my way to your podcast very recently. I was hoping you could explain a question about how this virus is possibly working. Why are the elderly more vastly impacted and children under 10 barely impacted at all? Why would this disparity exist? Do other viruses like this exist, Evan? I think some of that we would still love to know the answer to.
Yes.
Whether this has to do with physiological differences in the lung, whether this has to do with immunological differences, whether this has to do with exposure to other microbes. I think all of those things might be a part of this. I can think of things like chickenpox, where children have somewhat less severe disease than adults. And I even saw some really nice data on that at a conference once about it being related to a number of memory T cells that a person has that allow the virus to move around the body. But I would love to know the answer to your questions myself, Evan.
Yeah.
I think it likely
polio,
also,
right? Polio, right, Vincent?
Polio is more serious in as you get older, yeah. I think it has to do with immunosenescence partly, right? The older you get, the poorer your responses are, and maybe maybe your regulation is impacted, and that's why older people get, the immune pathologies, and maybe children are absent of those. But yeah, I think we don't know. And there's
respiratory syncytial virus, flu, those are in a way kind of special cases. That there you're talking about elderly, where it's clear that there's immune senescence that plays into it.
If you want to go back to West Nile, it's because the encephalitis that it's induced by the virus, the brain of an older person has nowhere to go, where the brain of a younger person has some little the sawtooth joint. So that
come now, Dixon. I'm
true.
Calling
It's
you
true.
out on that. No, the incidence of encephalitis is dramatically higher as age increases. We showed actually a graph.
No, I
of
meant
that
the symptoms, not whether you get it or not. I meant the symptoms.
No, I don't think
more
that's
old people die from West Nile than any.
Yeah, I know that. The incidence of encephalitis goes up.
Why?
It's because of the blood-brain barrier is different as you age. It's not because your brain swells and has nowhere to go, Dixon.
Well,
encephalitis happens before the swelling. It is, it's inflammation, right? I don't think it has any. I just don't think what you're saying has any basis in reality.
I wish that pathologist had shown up today.
What pathologist? Sorry. Let's get through these. Who's next? That was Brianne, right?
That was me. Yes.
Greg writes, "Thank you for the wonderful service you give to the world. My vote is NIH should fund the entire TWiX for all the free public work and benefit you provide to the citizens and research community as a whole. I'm certain your shows have motivated a large number of desperately needing students to follow a path in microbiology." And Greg wants to know about zinc, if it could be used. And zinc, has some antiviral properties. It's also it does something to the immune system, right? In theory, but mostly what you can buy over the counter in lozenges. This doesn't do anything as far as I'm concerned. So, zinc is rearing its head now again. People want it to be tried in combination with the failed hydroxychloroquine, but as far as I can tell, there's no reason to think that zinc would have any effect.
I think that there actually have been genuine trials with zinc that don't show any efficacy. If you read the label on Coldies, it says that it's been clinically proven.
Yeah.
But I would want to look up those studies. I don't think it's true.
I think the only data that is out there about zinc may be a very, very early time point. So if you take zinc an hour after infection, it may do something, but that's not what people are doing. You're not in an hour after infection when you're feeling bad and starting to wonder, maybe I should go to the store and buy some zinc.
Dixon, can you take Slims?
I can. Slim writes, "Hello. It's 3 degrees Celsius and cloudy here at 9:30 p.m. Sunday night in Montreal, Canada. First of all, great podcast. Thanks for keeping everyone informed with facts, especially in the age of troll, social media, and fake news. There have been articles in the past few years about zombie viruses emerging from permafrost reappearing due to global warming thawing. is it possible that the COVID-19 is one of these? And wouldn't it be something visible for a look into its genetic? Would this be worth a look into its genetic code to see if that's true? Basically, that's what he said.
Right. Well, the genetic code was determined in January, and it's not one of these viruses from the permafrost. It's a virus from bats.
Right.
Quite clear, but it's a good thought. But as far as we know, zombie viruses are not emerging from permafrost. Someone recovered a giant virus from permafrost. And so the theory is, as the permafrost thaws, maybe viruses will be released, but I wouldn't call them zombie viruses, right? They're just
viruses are zombies anyway.
You're a zombie.
Right? Undead.
Yeah, they'd have to be alive at one point to become a zombie.
That's right.
All right. So, Rich Condit, you're next.
Bob writes, thank you so much for being an intelligent source of knowledge regarding COVID-19. I believe you have many listeners that, like myself, found your podcast in an effort to educate themselves on the virus. I think the most obvious question we would have is, what the hell did you guys talk about before COVID-19?
Oh, you'd be surprised.
Virology is a big, big thing, dude. So you got 10 years of back episodes to figure out.
A lot of it.
There's no shortage of stuff to talk about. I'm guessing the pod was geared more towards academia. No,
not really.
Do you find the podcast more or less enjoyable now that you have to dumb it down a bit for morons like me?
Hey, wait a
minute. We've never changed anything. Thanks for the great information. So you go listen to back episodes, dude.
You're not a moron. If you're listening, you're not a moron.
Yes.
No, not at all.
Point.
No, I we've talked about lots of different types of viruses, and it was a lot of ways that we could discuss them and communicate them both to our listeners, but also get some information among ourselves and sort of show you what scientists do. That's what people have done all the time.
Brienne.
Yes. Ryan writes, I would like to point out a couple of ways that people can volunteer their time and resources, regardless of skill level. First, the Baker Lab has developed and hosts a video game called Fold It, where the objective is to predict protein structures and design proteins to have a given shape and sequence. Second, for those people who are not into playing video games, but who still want to help out, the Baker Lab also runs a project called Rosetta at Home. This is a program that you can install on your computer, which uses the Rosetta protein modeling suite to help predict the structure of a protein. And basically the way that this works is that by using many different computers distributed throughout the world, each contributing some predictions, they can get enough computing power to work on their models. And so Ryan is a PhD student in this lab and in the Baker Lab and at University of Washington, Seattle.
This is David Baker's lab at the Institute for Protein Design at the University of Washington.
Yes, and in fact, the next email from Niall says, to answer your caller's question about how people can help, also foldingathome.org. There you go. Let me take Amy's. Amy writes, I keep seeing assumptions that COVID-19 cases will fall in the summer, but can't we look at current data to test this hypothesis now? The virus is spreading worldwide, including in the southern hemisphere and equatorial regions. Do you know if anyone is testing this idea? What would we need to find in the data now to know if summer offers any protection? And it's a good point. And in fact, it was ripping through all places in the world, whether it was hot or not. And now here in the northern hemisphere where it's summer, it's still ripping through places like Texas and Florida and Georgia. So
California.
It's not behaving according to any seasonality. And I think that's what happens when the immunity is so low.
By the way, I haven't updated my Austin report recently. It's still the caseload. The hospital admissions are trickling downward slowly, which is good. The hospital cases are trickling downward slowly, which is good. Not fast enough for my liking, but nevertheless, it's a downward trend. The overall in Texas, the positive test rate is somewhere between 15 and 20%, depending on how you measure it. Not good.
Yep.
Dixon, you're next.
Eric writes, hello. Started listening regularly in January. I'm not fully caught up, so my apologies if this was answered in the most recent episode. I'm an engineer with some limited medical background. The general assumption has been that COVID-19 can be gotten. You'll have an immune response, and assuming you survive the infection, you'll be immune to it. Same thing for SARS. There was, as discussed on TWiV, news articles that were likely sensational and not true about reinfection. Influenza is a different virus, and the strain mutates and different strains travel through the human population regularly. A subset of colds are also coronavirus. Are we just getting different strains of coronavirus when we get colds, along with the other forms of colds? Why is SARS and COVID-19 considered one and done versus the cold? Is it just a function of the rate of virus mutation compared to influenza? I hope my question was articulated clearly enough. At least it was, because I read it error-free, I thought. Go ahead, Vincent. Sock it to him.
So there are 4 different common cold coronaviruses, and everyone gets infected with them every year. The immunity doesn't last more than a year, and you get reinfected from year to year. There's no mutation, as far as we can tell, that causes that, as for influenza. In the case of coronaviruses, the immunity simply wanes. And because, well, SARS-1 was come and gone in a year, so we really don't know anything about durability of immunity. But and SARS-CoV-2, we'll find out. But I think CDC just reported today, immunity lasts at least 3 months. For SARS-CoV-2.
Well, that's encouraging.
Because that's as far as they've been able to study it out. But I would assume that it's not going to be durable either, very much like the common cold coronas, in that you'll get reinfected. Maybe you'll have much milder disease, very much like the common cold coronaviruses. That's what I think.
I would agree. And I'm hoping that the reinfection results in a milder disease. And this is actually a very interesting letter because he's talking about there being recurring infections with influenza and recurring infections with coronaviruses, but the mechanisms are different.
Right.
In the case of the coronaviruses, you're talking about waning immunity to strains that don't change. In the case of influenza, you're talking about a virus that changes to evade immunity. So phenomenologically, it looks very similar, but genetically, it's quite different. Genetically and biologically, it's quite different.
Right. And how you would end up solving that is also quite different.
Yeah.
Rich?
Stefan writes, I am a clinical virologist in Tahiti, French Polynesia. All right. We ought to have a map with little pins on it. French speaking, but I listen to you almost every week, sometimes with some difficulty with my English. And I wanted to thank you for the quality of your podcast. Currently, taking a step back from the health situation that occupies everyone is a most welcome breath of fresh air. Your regular or guest speakers are always of rare intelligence, and it is good that the work you do is widely disseminated. Thank you. Kind regards, Stefan in Tahiti. Man. You ever been to Tahiti, Dixon?
Say again?
Have you been to Tahiti?
I have not.
Stefan,
would I like to go? I would like to go.
Yeah.
Go ahead, Dixon. Go.
I tell you what okay, Cookie writes. Your statement in your last podcast about trying to stop touching your face in an attempt to reduce the spread of viruses reminds me of the joke of an Australian horseman who started rubbing horse shit on his chafed lips. His mate asked, "Does that stuff really work?" "No, but it stops me from licking them." Yuck, yuck, yuck. Maybe we should put our hands in horse shit before, or should both to stop touching our face and to keep our diets.
Great. Publish.
That's interesting.
Why did you have me read that?
I wasn't. I was trying to ask you a question. This was Brienne's turn, but in fact, it's probably better that you read it than Brienne. Right, Brienne?
You spared her from spoiling her image, public image.
We couldn't have Brienne cursing, could we? No.
I can.
You can?
I can.
Oh, yeah, go ahead.
I've never. Actually, Vincent, you know what? That's probably microaggression.
Yeah, probably.
You know?
I'm sorry.
Implying that a female would
No, it's not a female. It's just Brienne. I just have never heard Brienne curse. That's all.
Okay, so I'm guilty of microaggression.
You curse for sure. My wife curses like a
sailor.
A sailor. Oh my gosh.
That's also stop.
Microaggression?
Yeah, just people curse. Let's put it that way.
Well, I've never heard Brienne curse. That's why I said. But you do curse, right, Brienne?
Yes, I do.
I apologize for microaggression. What was it that we heard in Texas? Someone opened the door and for a lady, and she said, "That's microaggression."
You opened the door for somebody, okay? And she accused you of microaggression.
In fact, he told that to Kathy Spindler, and she told it to us, and she was floored. She couldn't believe it. All right. In fact, I wanted to tell you something, Dixon, and that's and I can't remember, but Stefan's email where he said we're widely disseminated. We could be even more widely disseminated, but I guess this is about the best we can expect from a 3-hour unscripted radio show, right?
Yeah, that's true.
And
Rich asked me if I've ever been to Tahiti, and I said no, but I did know, and I do know, the former sustainability officer for the country of Tahiti. And he's on a quest now to find a place for the Tahitians when global climate change forces them out of their homeland, which is only an average 3 feet above sea level.
It's because of the water. It's going to flood the whole island?
Right. That's exactly right. So there's a project called Oceanics with an X.
Yeah.
And they're trying to build a floating country.
How many years is that from now?
Well, it's hard to predict, but I think it's accelerating. So I think whatever we could say yesterday was not true anymore. The ice sheet above Greenland is melting rapidly. The ice sheet in Antarctica is melting rapidly. So
Dixon,
it's a matter of time.
Will the water flood Manhattan at some point?
It sure will. It's only a matter of time. That's the point.
And it will. The whole island of Manhattan will be flooded.
No, not all of it. No. Not all of it.
How much? No. How much will be?
About a quarter of it. If you can think of what happened during Hurricane Sandy, that's about what would happen.
So we are now here in my building. We're like 100 yards from the.
Wait, what do they call where you are?
Fort Washington. Washington Heights.
Heights.
We're high up.
Washington Lows. Washington Heights.
Okay, so we're high enough up, right?
Don't move an inch.
I don't know.
Rockefeller's going to be in big trouble, right?
Well, all of downtown Manhattan, the Battery, all of Wall Street.
I guess Madison is okay.
Toast.
Madison is probably okay. There actually used to be, I'm looking right now and having trouble finding it, a Google Maps plugin where you could look at sea level rise and it would actually show you what areas were flooded.
Oh, yeah.
It was really interesting.
It's real, folks. It's real. So there's a book that somebody wrote. It's a fiction book called 2147. And it's about the year 2147 and what New York City will be like then. And this man imagines that the downtown area of Manhattan will become the new Venice of the world. And people will take advantage of the fact that it's flooded, and they'll adapt to the changing conditions. But that's
I wonder if that will happen, or if there will be a big seawall.
Nah, I don't think that's going to work. I know Germany is building one; they're already building one, so that they're convinced that this is all going to happen within several lifetimes.
Well, I keep saying that this pandemic is really just a warning shot.
That's right.
That the big one and the more insidious one is climate change.
You're right. So Galveston, forget it.
Yeah.
Right? Miami, forget it. Half of Florida, forget.
Yeah.
So that BSL 4 in Galveston is going to be
they're gone.
Defunct,
they're gone.
Right? Wow.
They're going to be underwater.
The whole university, the whole medical school.
The whole city.
Wow.
I'm going to be underground.
Yeah, so. We'll all be underground.
Recycling. We call that recycling, Rich.
If I found the correct site, it looks like Fort Lee would end up on a little island.
Fort Lee is doing well, and we're actually, that's true. This is very true. You could call that New Jersey Heights. If you wanted to say Washington Heights, we're at the same level that Washington Heights is.
I wonder if anyone's still listening at this point.
They're all going out and starting to build.
Doesn't matter because we're having a blast, right?
So into the ocean.
So Vincent, you got through all of the email that was on the doc, but there's another 100 pages somewhere, right?
Oh, yeah. So this document, we use a Google Doc for our show notes. And for months now, it's had 130 pages, and we've slowly whittled away. And it's hard because I add some new email. But for the first time, I think since February, we got through the entire doc, and now I have over 1,000 other emails to go. And, I have made it kind of a goal to try and get through all of them, no matter how long it takes and so forth. So I can tell you roughly, about 3 weeks ago, I started having all the TWiV emails forwarded to a new Gmail account just to keep them separate, right? And it's only been 3 weeks, so there's still probably 1,000 emails in my old Gmail account, but the new Gmail account as of today has 884
Good Lord.
Emails, and I will get through them. And many of them are the same subject, so it's not a problem. But, many of them are excellent questions. I'm just astounded at the number of excellent, outstanding questions from you listeners, so we thank you for that. We'll get through them. But that's it for today. And the show notes and all of that, the letter texts are at microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Dixon Despommier is over at trichinella.org and thelivingriver.org. Gracias, Dixon. Why do I use Spanish? I'm not Spanish.
I don't know, but I understood what you said.
Grazie, Dixon.
And I would say de nada.
Rich Condit's an emeritus professor at the University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time, and I enjoy my time with you folks. This has been delightful.
Brianne Barker is at Drew University. On Twitter, BioProf Barker. Thank you, Brianne.
Thanks. It was great to be here.
You should have said it's been one hell of a trip. Come on, use at least one curse word.
I have to say, listeners, these individuals, they just gave 2 hours of their time for you, and because we have fun too, but they're doing this because they like to do it.
We do it.
It's a privilege to, and not just TWiV, but TWiP and TWIM and TWiVo, all the other podcasts. People do it selflessly for you. So thank you.
No, I think the reason why people trust podcasts so much is because over time you're obliged to be yourself. And then they can tell whether or not you're genuine or not just by listening, because the tone of your voice and the way you behave and the way you react to criticism and that sort of thing, it brings out all of the personality traits of that person. And they love knowing who you are. And on television, there's no chance for that whatsoever.
No, you don't have any time. No, I agree. I think that's a good point.
We have the luxury of time. This is all true.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.